Yo. turning down the volume there a little bit, you guys can start to hear actually that clipping on the hydrophone. That's that interference there. Is if you're the gain on these hydrophones and cats tags are actually kind of sensitive. So usually, if we'll talk a little bit about deployment ideas towards the end as people have questions for this. But if you're using cats tags specifically, you sometimes get this clipping. Yeah, and we're getting some comments on like what that kind of sounds like. But basically, where there's like the tags are meeting their maximum value. So usually I set the gain to really low on this because you don't get that clipping. But I like this clip because it kind of shows the power of some of these tools for biologging, because we have basically like multiple animals doing multiple things and kind of recording the data from this one animal, but also watching other animals go. Sorry, I heard someone else jumping in there. Is there another comment?
That might have just been me agreeing, Dave.
Nice. Give people another minute here. But so yeah, and so like one of the things to think about is just another thing to think about with your data, right? Like what are you setting your hydrophone data to, and what are your purposes for? Like if you want to detect really far away sounds, you want really sensitive hydrophone, but then you might get clipping at these really high, fast locations. And so if you're trying to use it to detect speed, that might not work for you. But then you have this alternate speed method, which is the accelerometer jiggle. So you have these multiple things today to use, multiple things to think about. All right, it's great to see everyone here on this last day here. Really great effort. It's really heartening to see people push through. We saw a lot of people, pause this so it's not, I'm not talking over a minke whale. It's really heartening to see everyone show up here. We've had a lot of people come up with no MATLAB experience to start with, and now they're making PRH files. So that's really great to see everyone. Thanks for everyone's high energy for this whole process. I know this can be a really challenging thing to learn online, so it's really cool to see everyone really make a lot of progress over the course of the week. So really great. Towards the end of the session, we will send you guys out some feedback links. It'd be great to get, this is mine and Will's first time teaching online courses. So I would love to get some feedback on our teaching styles and on the workshop format. And I'm sure you guys have ideas about how we can improve it for future kinds of workshops. So we will we'll send you guys that out towards the end. Today, we're gonna do a couple of things today. We're still trying to look at a little bit about like other tag types. So we're gonna have some guest speakers today talking about other tag types and how to like adjust our tag formats for that. And I'll show you a couple of examples of where people are getting stuck right now and how we're going to fix things in the future. And so just kind of give you an outline of the day today. So what we're going to be doing, we'll just do a little bit of introduction right here about like where we're at and where we're going. And then we're going to come back to using Boris a little bit to do a little bit more wrap up for how Boris is used for science. Sarah, Nicole, and Emily are coming back to nicely agreed to come back because we ran out of time a little bit yesterday to talk a little bit more about Boris. Then Taylor Chappell's going to jump on and talk a little bit about different attachment modalities for CATS tags and some other species you can use CATS tags on. James is going to jump on and give us a little bit of information on using long data sets and kind of adapting CATS tools to work with other data sets and some of the things to think about there. Then John Kalmakidis is going to come on at about 11:15 and tell us just a little bit about his experience with tagging and thinking about why and when and how to use tags and different kinds of tags that are appropriate for different purposes. I think it'll be mostly cetacean focused, but it should be pretty rad. And then I think Ari Friedlander is going to jump back on around the same time, and we can ask a little bit of a panel questions if people have some time. Then we'll kind of do our final version here, our final end motion, will be our last hurrah. We'll be stitching videos together with the data, and I'll show you the procedures to do that. And you can run that. It takes a long time, so you can run that though on our example data set if you'd like to try that. You will need a monitor that has a sufficient resolution to be able to do that, but you can start thinking about how to get the tools you need to do that. And I'll show you some of the examples for that. Then Will's going to kind of wrap it up with a little bit of like, okay, what are the next steps in future processes? And then at 1 o'clock, full open bar for everyone. Come on by. Just, you know, if you're anyone who's taking the workshop locally, you can just come on by, open bar, all good there. So come on, check that out. I'm going to show just a couple of bit of like where we're at. And then we'll jump into today's stuff. So swapping around here. So last night, As of yesterday, everyone should have, we had a lot of people working through this yesterday in the afternoon session. You should have basically a well-populated folder now with all of your data, the critical files being this PRH file, and some of the other files that I don't actually see here. Let me show you another example. We'll go to our main example here. When you're finally done, you're going to be starting to organize this. You can organize this into your tag data folder. I like to have one folder that just has all of my separate data. So you should have this PRH file. You should have these this ACQ file, which is basically a text file that has basically, if you like to use just normal text values, ACQ stands for acknowledge, which is an older software you can download that can just look at text files. And then you have an NC file, which is the highly portable format that you can use in R or anything like that. So those are all outputs from your file structure last night. I know a lot of people had a couple of errors cropping up that I want to just highlight. One of them is just a folder structure error. So one of the two first things we'll commit to, and I haven't confirmed this with Will, but basically we'll try to, by the January 1st is our commitment to try to make sure all the tools are kind of static and come up with some of the changes that people have highlighted this workshop that need to be made to make this a more usable tool. So once 2020 is in hindsight and it's gone out of the window, everything should be good in 2021. So we'll have this up and ready to go, including so two of the main errors people are coming up with. One is a folder structure error, and Will kind of talk us through some of that, including like how to find all your quick look files and how to know where those things are. So we'll talk a little bit about folder structure, but we'll also do our best to make that an optional feature so that if you have it all organized in the way that Will's talking about, then it should be really easy to find all the files you need. And if you don't have it that way, then we'll just include better prompts to look for that instead of throwing errors so that you can find the files that you need to be able to create the files that you're looking for. The other error people are coming up with, especially when you're using your own data, or if you created your own truncated file, if you're not using the example truncated file, but you're created your own, then people have been coming up with this error that looks like this. Basically, the critical error in here is that it's trying to make a plot here, and these two functions, depth and temperature, have different lengths. And that's bad. It's not going to be able to plot that if you have different lengths. So, and that comes from the way that we decimated. I introduced a new decimation feature here, and you can see it right here, this decimate M, to decimate all the files to different lengths, and they're coming up with one off. And so I know that we've known that it's an error since Wednesday, but I haven't, I want to make sure I fix that in every location where it's a problem. So I haven't fixed that yet in the script. So I want to make sure and do that really thoroughly. So we will come up through and do that. Those are my main intro pieces for today. I'm going to pass this over to Will, who's going to talk a little bit about the forum and some of the folder structures before we pass it over to the Boris, Sarah, and Emily and Nicole. I see a bunch of questions coming in, Will, too. Feel free to jump in with those if we need to take a second to think about them. Let me take a look.
Okay, I'm muted. Sorry. Yes, give me 1 second here. Okay. Yeah, so just to maybe go through it a little bit. Also, thank you to Anna for going through and sort of giving some timestamps on the last few days of talk. That's going to be really nice. I'm going to actually put that on the website here while some people are talking. But yeah, so just really quickly, let's go through kind of like file structure for, how we kind of set up things for the quick look, which is going to be this last phase of the PRH process. And what I'm going to do, let's see, let me just open up. I'm going to share. And so I'm going to kind of show you guys just really quickly, kind of like, this is one of the hard drives that I have that we actually took out into the field. And so some of this data has been processed and some of it hasn't. But just to show kind of this file structure. So we have this cats folder. Typically, it's how we set this up, with that being like the main piece. And I'm going to jump back and forth a little bit here between that and the this page 8 of the wiki. And my goal, and I'll talk about this a little bit at the end, my goal is to sort of update and have more on file structure, because it seems like that's an area that people want a little more information. And so I'm going to try to blow it up a little bit and have it be its own section so that people know kind of like not just figuring out the file structure at the end, but actually knowing from the start where you should be putting your files and what you should be naming them and things like that. So we're going to kind of be working through and figuring out like where we need to put all of these files that are going to go into this quick look. And I'm going to kind of start from the top here. Again, kind of like it says, this highest level is going to be this cats folder. What I'm going to do is I'm just going to put this on one side and keep this on the other. And so within this cats folder, again, kind of like it shows here, we have our calibrations folder, and we've had a lot of tags over the years. So we have a lot of different calibration files. So once you've made this calibration file, again, kind of like we talked about on Tuesday, that file is good to go. You don't really need to redo it for that tag, barring something sort of catastrophic with the tag or something like that. But you sort of have them all in here. One thing you do need to make sure, and we actually ran into an error with somebody the other day, sort of trying to figure this out, working with a different tag. You need to make sure that the file here is named with the tag number from your deployment. So if you have tag number 1, you got to make sure that sort of like the tag num variable in your PRH lines up with this, or in your data, sorry, in your data lines up with this file name. Because otherwise it'll throw an error and it'll basically tell you that like your tag number does not align with your calibration file name. Okay, so we have that there, and then sort of more importantly, we have these two folders. So we have our tag data and our tag data raw. So we're going to go into tag data, and I'm just going to sort of choose one of these deployments. And actually, that's not a great one. Yeah, let's look at this one for a moment. So we have our raw folder here, and typically the only things that go in this raw folder are the things that come directly off of the tag. So for us, that's the video and audio, the CSV data, and then the 3 extra files, which are the bin, the .bin file, the .txt file, and the .ubx file. And so that's the only thing that we typically keep in here. Everything else that gets created, so the mat file when you import, the movie times file when you run the first cell of the cats PRH script, the truncate file when you run cell 3, all of these files, your header file, your GPS hits file, everything goes outside of that raw folder. And we typically try to keep it in there. And as you're working through, just to kind of show, basically everything gets created into this folder, right? So your PRH that comes out gets created into this folder. Everything is sort of here. And so, if you want, you can keep it all in that folder. What we tend to do though is we kind of have it split between sort of like we have our tag data raw, which is like, this is where I'm working on stuff. And then we have our tag data, which is a little more finalized. So I'll show you guys that really quickly here. And let me know on time, Dave, if we need to move on. So this is our tag data folder. So this is for that same deployment that I just showed you, but it's sort of like the finalized pieces that people might use and actually want for various analyses. So in here, we have our PRH file, which is really the most important piece. We actually have our videos in here. So these are the finalized videos or semi-finalized videos. We have our NC file in here that Dave talked about a little bit yesterday. We have a lunge file, which is something that Sherelle helped to make and other people in the lab, where it actually tells you where all of the lunges are in the deployment, all the feeding lunges. And then in terms of the actual quick look, sort of what you're making here, what you're going to need is basically these two different folders here. So you have your quick look folder and you have your pics and vids folder. And so what the last step of the CATS PRH program is going to do, that script is going to create the PRH, and it's going to look in these different places to find the right information. So first of all, it's going to look for your PRH. So you need your PRH to sort of be in there in this folder. And then it's going to look for your pics and vids. Let's see. All right. So it's going to look in here. And so, you know, I have some, you know, some photos of the fluke of this animal. So this is sort of like an ID shot, one that may be a little bit better. Yeah, you know, not great. And then there's just sort of some different things in here. And basically what we do is we take whatever we have and we try to make two files in here. One is the ID file and one is the tag file, or the ID image and the tag image. And so for ID, we basically just take the best, either it's a fluke shot or for, you know, depending on what species you're working on, maybe it's a dorsal shot, or if you're not working with whales, maybe there's another photo you have of like the ID of the animal. And we sort of just zoom it in and try to, you know, increase the contrast a little bit so you can really see it. And then we save that as this separate file, which is ID and then underscore and then the file name, or the deployment name, excuse me. And then we do the same with the tag. So for this one, this is actually a good example where we didn't really have like a drone shot, or we didn't have a good photo from the boat. So really what we had to do is go to the deployment video, which we had on a GoPro, and just take the best screenshot that we could from there that sort of showed the tag. And the goal of this is to just sort of show, you know, approximately where on the whale did the tag go on, right? Did it go up forward on the blowhole? Did it go near the dorsal fin? Was it back near the tail? Was it on the left side, right side? It just provides a little bit more context when you end up creating this quick look. So
is that a good place to transition? Or sorry.
Yeah.
Yeah, just to transition into the next phases. I think that's a good bonus there to have wrapped up the pics and vids and the quick look there as like as an idea. I wanted to just highlight that's the quick look there that Will was highlighting. That's basically, it's a cosmetic piece. It's another piece to like make you organized. But a lot of what we've been showing you guys are just like the raw data pieces. Everything up until that step is like, how do you create data? So when we've shown you like things like figures that are not that awesome, like they're not trying to be publication quality when you're showing, you know, figures like this or even this like map figure or whatever, right? Like we're not trying to be publication quality here, but that quick look is just the idea of like helping you organize your data, and you don't have to do that to run with your data. You can come up with a whole different format. That's just one way that Will is showing you that. Are anything else, Will, to show at this point, or are we ready to move on to the next step?
I think we're good to move on. The only other piece that I might mention quickly is just that you're sort of going to be doing the same thing where you're taking videos and you're taking some of those videos and you're also creating some of these files in here. So you're creating these two from that KML file, so in Google Earth, and then you're creating these two from the actual tag video. So you're going in and you're just finding sort of like, you know, one or two interesting screenshots from that tag. And then, yeah, once you have those and you have them in this sort of format here, you should be good to go to run that if you're interested in creating that.
Awesome. And I think we'll circle back to at the end to kind of some of the other file structure and ideas. And we'll circle back to some of how to use the GitHub and how to use the wiki and things right towards the end to like talk more specifically about how to like move forward from here.
Yep.
Awesome. Cool. With that, I want to turn it over to back to Emily and Nicole and Sarah to kind of continue a little bit with thoughts about how to use Boris to help, like once you get these video files that we'll create at the end of the day today.
Perfect. Awesome. I'll go ahead and share the slides from yesterday. Sweet. So I hope that everyone enjoyed kind of that first look into Boris yesterday, and seeing just kind of the few things that you can start to do with it. And then for the next 5 or 10 today, we'll just go over, Nicole and I will go over some of the studies that those in our lab have already done using both the CATS tags and Boris. So the first study is a behavioral ecology application, and this is a study that Ariana and Sarah in our lab have done. And they've been utilizing Boris to audit CATS video tag data collected in Monterey Bay and Stellwagen Bank. So the footage we worked with yesterday for the sample exercise were 2 video clips taken from the data that they've been working with. They used Boris to record and evaluate the observed behavior of humpback whales, along with the relative abundance and behavior of any observed associate organisms. They developed an ethogram based off of a template that our lab uses for general auditing, but refined it to include specific variables and modifiers that they were looking to evaluate. This included surface and dive intervals, lunge feeding, bottom feeding, bubble net feeding, with modifiers for prey type and estimated abundance, interspecific interactions with modifiers for abundance and behavior. And the goals of their project you can see here. They wanted to analyze similarities and differences between observed feeding styles of the focal humpbacks within and between the 2 locations, to compare and potentially link those findings with the presence, abundance, and behavior of any observed associate animals. And they wanted to use their comprehensive findings to provide hypotheses and suggestions for future research projects. So a little bit more about this example. Between the 2 locations, Sarah and Ariana observed different behaviors from the focal whales, along with the presence and type of associate organisms. In the Monterey Bay footage on the left here, Sarah has monitored humpback whales utilizing lunge feeding, while the focal humpbacks perform these feeding lunges independently. Unlike bubble net feeding, Sarah has been observing other humpbacks and large clusters of California sea lions present during these feeding events, potentially benefiting off the behavior. This snapshot was taken from the Monterey footage and demonstrates the extent of the clusters of both sea lions and humpbacks that are found together off the coast of Monterey. Sarah has observed that the presence of humpbacks appear to help disperse the large prey clusters and allow smaller species like the sea lions to predate on the then scattered prey. And then on the right, unlike the whales in Monterey, Ariana is noticing that the whales in Stellwagen utilize 3 different types of feeding strategies: bubble net feeding, lunge feeding, and bottom feeding. And she's also noticing an overall minimal presence of interspecific associates in general, with most of the associate organisms being interspecific organisms that are displaying coordinated feeding or traveling behaviors. In this snapshot from some of the Stellwagen footage, you can observe several seabirds hovering above the surface as the focal animal and another associate humpback surface during a bubble net feeding event. The second application is an environmental assessment, and I'll turn it over to Nicole to explain this study.
Yeah, thank you, Emily. So another use of this auditing feature, in addition to the behavioral stuff, is more related to environmental assessments, as well as kind of describing habitat use of some of these focal animals. So some of the goals of this could be to qualitatively observe the in situ environmental conditions. So what our lab has used it for so far is sea ice observations, but I think it can also have broad applications to any number of other environmental assessments, as well as to quantify the environments in which these animals spend the majority of their time. So some of these questions that you may be looking into can be related to just simply describing the real-time and like fine scale resolution of the environmental features where these animals are occurring and performing all these behaviors that we're trying to look into as well. In addition to quantifying these environments, which I'll talk about a little bit more in further detail in the following slide. But yeah, you can develop methods to continuously quantify these environments and then relate this back to habitat use and as well as the behavioral observations. And all in all, it helps facilitate just overall understanding of how these animals utilize their environment and kind of determine whether it's environmental features driving these animals' behaviors or if the animal behaviors are being adjusted to the environments in which they occur, which I'll talk about more a bit in the following slide if Emily, you want to switch it over. Thank you.
Oh, it's if it lets me.
No worries, thank you.
Been locked out. Okay, there you go.
Okay, thank you. So one of the cool projects I've had the pleasure of working on is developing this method to quantify the sea ice environments of Antarctic minke whales with the tag data associated with that. So in this top left-hand screen are kind of examples of the ice bin classifications we are able to develop. And the associated like percent concentration are shown above each one. And these are just snapshots as we audit the videos. It's kind of an interval of each surfacing event that we're looking into for each tag deployment. And so for each tag deployment, 2 people are assessing these video footages and then later compared. And if you do want to learn more about the nitty gritty of like the actual methodology we developed to quantify sea ice, it's in this paper that's shown in the bottom left-hand corner that was just published this year to describe in more detail how to actually quantify the sea ice environment. But what that can be used for in future research, these 2 frequency histograms at the bottom are for 2 different tagged animals. And it just kind of shows like the contrast between even just 2 animals of the same species. So for instance, like the one on the left spent more time in relatively open water compared to the one on the right, which has quite a more variable ice environment that it utilizes. And then this is kind of poor resolution, but on the left-hand side here, this colored graphic, this is a whale track, and at each surfacing event is a corresponding dot showing which ice classification it falls under. So this can also just be used to kind of describe how different animals vary within like a time frame, the different ice environments that it might interact with and what behaviors might be coordinated with those movements. So it's just another lens into looking into what features are driving these animals and what might be important to them. For instance, with the minke whale stuff, they're very they're ice affiliated, and before some of these tag deployments, we were uncertain about how much time they spent in these sea ice environments. So by looking into the video footage and quantifying how much time these animals spend in these environments, we kind of get a better understanding of their dependence on these environments and how that might be changing in future climates. So it's a powerful tool in that regard as well, just in terms of looking into real-time lenses of what these animals are experiencing. So that's what we've been able to accomplish so far, but it has broad applications to other environments as well. I'm curious to see where it can go from there. So that's all about my work. I would send it back to you, Emily.
Awesome. Okay. Yeah, so the last application is a physiological application. And this window is now probably fairly familiar to everyone after yesterday. And this will be the final example, which is the study that I led, which primarily used Boris as a video analysis program. So here I didn't actually use too many of the auditing features. I used Boris to create a new method in order to study ventilation. And I think Boris is a great way to view and sort through your video data because of that auditing feature where you can create a library of the events or behaviors that you're specifically interested in addition to replaying and capturing very specific moments in time within the video. So for this project, I used video recordings from the cats tags placed directly behind and in line with the nares of the humpback whale or minke whale, which is the blowhole. And then videos were broken down and viewed in frame by frame mode, which you can see in that red circle kind of along the top. And I think frame by frame mode is a great feature if you're trying to pinpoint kind of an exact moment in time within your video. And in this case, that exact moment in time was when the animal began to exhale, inhale, or the transition between the two. And I also used the screenshot function within Boris to look at how much the nares expanded between frames throughout a given inhalation. So using these methods within Boris, I then calculated the how the area within the nares and the duration of the inhalation changed between breaths and breath types. So the first breath taken at the surface, any of the middle breaths kind of taken while the animal's continuously surfacing, and then the last breath taken before the animal then dives. And in this study, I found that that last breath taken at the surface both had the longest duration and the largest nares expansion. And some of our results also suggest that the duration of that last breath at the surface may have been influenced by the following: dives, total lunge count, and duration, which may indicate some pre-planning prior to dives as observed in some other diving animals. These video measurements have important physiological applications and could change our ability to understand and estimate the energetic costs of different behaviors. So continuing to use Boris in the future to study metrics such as inhalation durations could be a critical tool for estimating ventilation and lung volume in wild baleen whales. And that's it.
Yeah, that's so good. We'll take a couple minutes for questions, and then before we turn it over to Taylor, does anyone have any questions for these guys? Just a really cool, powerful tool to do some of these audits and video analysis, and people have really been turning it into turn these videos into like things that we never expected to see, right? Like the aerial area of an Aries, and just turn that into data is pretty cool. So really cool.
I did have one question on the measurement part. How do you ground truth Boris for like size? Are you just doing relative size in each video, or can you actually get like the measurement of the Nares opening itself in like centimeters, say?
Yeah, so while I was using Boris, I just used the number of pixels actually within the Nares. So it was just kind of this, I guess, relative size between within that whale itself. But the super cool idea in the future would be to use some drone footage if the camera technology improves, where we could actually get the exact centimeters or millimeters or however much that the Nares are actually expanding to hopefully put some real measurements to that.
Cool, thank you.
Yeah.
Awesome. There were some versions of CATS tags too that we started to think about having two cameras on them. So you could potentially do a stereoscopic measurement of things in situ as well. But that would still be a future project for someone to work with CATS on. That would be pretty sweet. Cool. Thanks so much, guys. That was super cool. That's really neat to see those things. Thanks for doing that. On the vein of like things that I wish I could do and be as cool as, there's all that stuff, and now we're going to transition to Taylor, who does a totally different species. For those of you guys who've been waiting for us to get off the whales, this will be good for you guys.
Cool. All right, I will get started. Thanks, Dave. So first, I want to start and say thanks to Dave and Will. Are you seeing that? All good?
We're seeing that, and I've been doing a terrible job of introducing people. And so Taylor is a professor at Oregon State University, has been working with sharks all over the world, and so he's going to talk about this work here. And you can introduce yourself a little bit more as well.
Cool. Yeah. Thanks, Dave. So Dave and Will, thanks so much for this this workshop. Basically, a little bit of my history. I started working with the cats, Nico and Peter, back before they were really cats in 2013, and having these tools would have saved me years worth of banging my head into the computer. So for all of you just getting started, this is an awesome resource. And it's great for me as well. So yeah, I'm going to go a little bit different. I'm going to go away from marine mammals. So that's why I sort of titled this, Not Everyone Loves Marine Mammals. And that's just for you, Dave. So because Dave and I always give each other a hard time about our different species. So I'm going to talk about some of the attachments that we do for sharks, and how the data look a little bit different. Let's see. So this goes back to the same ideas that Dave sort of spoke about. Our historical understanding of my study species were from these really brief interactions at the surface. And so the idea that you titrate an animal's day down to this 30-second interval and then interpret the rest of its day from those times, it really doesn't work. It's like seeing you at the coffee shop in the morning, getting a cup of coffee and saying, I can describe everything that you've done over that 24-hour period. So it doesn't work that way. So these tags have been really invaluable for us to really understand what the animals are doing. And so the tags that I've been deploying, and those that work on some of these animals that you have to clamp tags onto, we use a fin clamp, which is basically like a clothespin that you would put on your clothes. And so if you look at this figure, this is one of the old first generation tags in the middle there. So it's got the camera body that fits into the orange float housing, and then you see the metal sort of clothespin piece that's attached to the attachment pole there. And so as a shark goes by, or whatever animal that you're working on, you clamp those on. And then the one in the top right corner is a stomach tag, which I'll talk about a little bit. And I realized I should have also included tuna tags that we've been using to clamp onto tunas, and that's a little bit more invasive, where you're actually drilling through the fin, and I completely forgot to put those in. But anyway, so that's how the tag works. One thing I wanted to sort of stress and mention is that there's a lot of, we're talking about the analysis part about programming the tags and then getting them back, but there's some steps in between that are really important that obviously there's not enough time to cover all these things. But one is just the simple, like making sure that your package floats when you get it. This is Adrian Gleiss, and if you don't know Adrian is an incredible scientist and has really been on the forefront of this biologging stuff. And this is our work in South Africa, and it took us a few days just to get the tags to float right. We got them out of the package ready to go, went to go put them on, and they all sank or they floated sideways. So just little things like that to make sure that we think about before we deploy. Another thing is that we talk about putting the tags on and then how we analyze the data, but there's this big step in between, which is actually retrieving them. And so one big consideration is what the conditions are like when and where you're deploying. So not every day looks beautiful like this, where it's flat and calm. Some days look like this. And so this is a searching for a tag in Central California on a 50-foot swell day, which was not super ideal. We've also had to do things like go up in helicopters. You can't really see, but behind me is the receiver antenna trying to find it. So a lot of different techniques. And actually, I think I would suggest that maybe some of us that are familiar with these different techniques, especially James going all over the place trying to find tags, is that we put together maybe a document or a short paper that outlines some of the strategies to find tags. Because I think that is one of the hardest parts, is actually getting these tags back. So you all know if you put a tag on, you don't get anything out of it unless you get it back. So that's a big part. So just some of the other considerations. And I think John later will talk about some of the ethics and things about animals, which I think is a really good conversation to have as well. So back to the tags that we use. So the tags that we put on get clamped onto the dorsal fin. And this is some work in South Africa. So you can see the shark comes up to the side of the boat, and then as it swims by, I clamp the tag on. And so the idea with this is very similar to putting them on marine mammals. It's trying to be low impact and low risk to the animals. This just gives, I thought this was kind of another cool view of the process. And so this is a shark that obviously already has a tag on it, and you can see, so we can actually see tail beat frequency. The shark comes on, then we clamp a second tag on it, and we were doing some different work. One was just a camera, the other one was a logger tag that had speed sensors on it. So the idea is this animal goes right back to its normal behavior afterwards. So we put a ton of these tags on different animals, and I put this figure in, and actually these numbers here are significantly higher by this point. But I put this in just to show sort of a difference in the size class of these animals that we work with, and just something to think about when you're talking about the actual attachment. And so the clamp sizes and things like that is that you've got about a 9 or 10 footer on the left, and then that's about a 17 or 18 foot animal in Central California on the right. So even clamping tags on to different size animals, even the same species, can be very difficult because the size of the animal changes, the width of the fins and things like that. So just other considerations to make. And I want to show this. These are the sort of the raw depth data, and I wanted to show that because I think there's some of us out here that don't get these beautiful periods up at the surface that Dave and Will were showcasing for the calibration stuff. And so it is still possible, as Dave said the other day, it is still possible to calibrate them even without those. And so you can see there's no real resting point in this tag. We're still able to calibrate it. So if your data aren't as pretty as a marine mammal, there still is hope for getting good data back. Another thing I wanted to mention is obviously the scope of this week. We don't have all the time in the world, but another program that is really important and has been really powerful for analyzing these data is called Igor Pro. And so you might have heard that or read about it in some of the papers you've looked through, but that's something that hopefully we can put together a workshop to teach more about that as well. So that'll be sort of like the next steps after you get your data really calibrated and looking pretty. in really solid in what Dave and Will have showed us this week, and then doing some analysis in UR Pro. So with the sharks, Dave, we have these also have speed sensors, and we talked a little bit about this. The problem with sharks is that their average speed isn't quite, I don't think, quite high enough to do some of the speed measurements that Dave talked about. And so for us, we put these little speedometers on them that have a magnet in them. So when it rotates, the magnetometer picks it up. So we do a little bit of a different calibration on it because of the slow speeds. And some of you that use work on slower moving animals, this might be an option. But again, they're pretty, they can be tricky in order to calibrate right. So I'm going to go through just a little bit of the data in some of the things that we've found and the type of data that we pulled out that, again, is a little bit different. So these are just some speed data from a few of the sharks. And these are 3 different sharks on the right side. And I just wanted to point it out that, again, the data look really different than what you've seen over the last week. And so if again, just to show that there's a wide variety of data that you can get back, but it is possible to calibrate these the same way and really get the same type of results out that Dave and Will have been talking about this week. And this is just some of the video data that I wanted to show. So this gets back to sort of our conversation yesterday about picking out these different these different behaviors from the video and looking at signatures within the data. So this is a shark that breaches out of the water, that really characteristic behavior that you see on TV all the time. And from that, we're able to pick that out from the data signature and then go back in. And what was cool is that we figured out that these animals were going from a tail beat frequency about 1 every 3 seconds to up to almost 8 tail beats in a single second, which is really incredible for a couple 1,000-pound animal that's moving that fast. And then actually this fits in really well with what we were just talking about in terms of using the video to figure out sort of how they're interacting with their environment. And so I'm not necessarily looking at sea ice, but we're looking at how these animals utilize their habitat. And so this is a white shark that's actually hunting in the kelp. And so you see the seals popping down and blowing bubbles right in front of the shark there. And so this is a behavior we've never known to occur. So we've never actually seen this obviously from the surface, and we thought that sharks avoided this. And so this is the type of information that we're pulling out. So the same way that that you're categorizing sea ice on the surface, we're using the same exact process to categorize kelp coverage in the view. We were using a product or a program called Solomon Coder, which I think is very similar to Boris. And I'm actually really excited about Boris because Solomon Coder is no longer supported. So Boris, I think, is a great option for looking at these behaviors. And again, we pulled it out and saw that these sharks are actually utilizing this habitat to hunt in. And then this is a pseudoplot, very similar to what we just saw with the whales. In the green, the black line is the movement of the animal, and the green is inside the kelp. And we see that the only time these events of seeing their prey happens is inside that kelp forest where those red dots are. So again, very similar to the analysis we just saw with whales, but something similar that we're doing from the data with sharks. And this is just a timeline of 8 different sharks that shows those events basically. So this mixes the 2 different analyses that we've seen in the last 2 days, looking at the event analysis and then the Boris style of visual analysis. So basically what was really cool about this is that we were able to use the video to look at predation rates of these animals. So our sharks, these mindless killers that go around eating everything, we figured out from about 450 hours of video from populations across all around the world, there was only a single predation event, which is kind of crazy. And so I like to actually show people the predation event, which is it's here, and you can all be wowed and amazed by the first ever capture of a white shark natural predation event underwater. And that was it. It was a hagfish, for those of you that aren't familiar with your small slimy marine creatures, which is pretty hilarious. So the only time we've ever caught out of hundreds of hours of video is to catch a white shark eating a hagfish, which is pretty interesting. And then I'm going to talk really quickly about another tag that we've used. So that's the typical CATS biologging tag that's on the fin, and we wanted to look more about some of the behavior, the feeding behavior of the animal. So we sort of incorporated a satellite tag from Wildlife Computers, with one of those, the daily diary tags, which were basically the precursor of the cat's biologging tag. We put those together and we fed it to the animal. And the cool thing about white sharks is that they are warm-bodied. So when they eat, they increase their temperature, basically for digestion. So we can look at the stomach temperature of these animals and figure out if they've had a successful feeding event. So if they go to eat something, the stomach gets cold from the ingestion of cold water. And then if it's successful, the temperature will elevate up really high while it's digesting. If it's not successful, it'll just go back up to its resting potential. And we can pair that heat signature with the biologging signature, the accelerometers and things. And we can see if this was a predation event and if it was successful. So on the right side, you see that little box that says feeding. You can see the temperature drops down and goes back up. So we know that was successful. And then we look at the figure on the left side, and on the top is the depth signature. So we can see the shark is at that moment is basically rocketing up the surface. The acceleration in the B part of that figure shows that this shark is increasing its tailbeat frequency. And then something we haven't really talked about, which is ADBA, which is this overall dynamic body acceleration, which is basically a combined measure of activity or energetics. And we can see that there's this big peak when the shark goes up. So this is a shark that's actively pursuing a prey at the surface, bites onto it, and actually did successfully consume something. So it's a different type of tag that we're using. But again, these data without the video capabilities can still be processed in the same way that, and calibrated the same way that Dave and Will have been teaching us all. So just to show another sort of result from that, it was at over about 1,000 hours of stomach tag data. There was only one single successful event. So really gives us an idea of the foraging rates of these animals, which is pretty cool. I'm happy to talk more about if anybody does these types of attachments or wants. We also take sharks on the boat and drill through the fins and attach them that way, which I've forgot to put a picture of. But if you have any questions about that stuff, feel free to reach out to me, or I'm happy to I guess lead you to someone else that does similar species or whatever. So it's a different type of attachments, but again, the data come out really valuable for all of us. Cool. Well, thanks a lot.
Thanks, Taylor. That was awesome. Yeah, we all can. I know that I get stuck in my okay, my study species. Here's what I need for my study species, and so it's really fun to get some different insights from different animals. So thank you so much. Are any questions for? I see one question for you, Taylor. Is when the tags are on the fins, do you see increased noise when the shark is at the air-water interface at the surface? Are you able to get good accelerometer data there at the surface?
Yeah, so the thing is that they very rarely get their fins out of the water, and we do see a little bit more activity. So if you look, there's a paper by Yuki Watanabe where basically at the surface you're in that last meter of water that it gets really junky because of that barrier there. So it does get a little bit wonky. It's hard to tell. Basically energetics once you get up that high. I should say that at one point because the sharks were sort of getting scared away by this big orange thing at the surface we're waving around to try to put on them, is that we were wrapping the tags in those black biodegradable dog poop bags. And so that when the tag, so I should say that the whole clamp falls off. It's got a corrodable link. The whole clamp falls off the fin, and then the camera tag falls off the clamp as well. There's corrodable links on all those sections. And so when the tag came off the clamp, basically it let go of that plastic bag, it biodegraded, and then the orange tag was floating. But when the sharks were swimming, that bag, the vibration of that bag was enough for us to be able to pick up the signature of it. So even though we couldn't do movement because the sharks weren't swimming fast enough for the tag to wobble, with that plastic bag on, you could hear it and see it a little bit in the acceleration data.
Like the dog ears wrapping along in your car window. Exactly.
Exactly. Except it was a dog bag. So
I'd add to the surface water interface. I did do some work with whale sharks, which do spend a lot more time at the surface, kind of filtering along in there with different kinds of tags, with some open tags. And they, that is a problem. That is certainly a problem. When the tag leaves the water, you have a totally different motion. You might have a lot more fin wobble, or you just might have a lot more integration and error. And so that when you're trying to measure things like ADBA, if you have different air-water interface boundary problems, you might have to be really careful about doing those integrations differently.
Yeah. Well, one thing I wanted to say is that, and I mentioned this the other day in one of our breakout sessions, is that we've been working, developed for anybody that puts them on with a pole. We've been developing a new sort of like adjustable technology for the tagging pole, and I couldn't find my phone to take a picture of it, but if anybody's interested, I'm happy to send photos of it. We basically machined some pieces that use bike brake cables, so you can manipulate it in any directions at the time. So you're not, those of you that have used it, you sort of get this static pole that comes with the tags. But if you want something that's maneuverable, I'm happy to send those along to anybody. It just makes it a lot easier when you have a dynamic animal that's coming in to tag.
Cool. You have some interest there, I think, Taylor. So you might have to, you guys reach out to Taylor. I think, we'll share your contact information later on, Taylor, maybe, is that all right?
Yeah, I'm just taylor.chaplett.oregonstate.edu. Or you can just look me up. I'm at the Big Fish Lab at OSU, and all my contacts there. Happy to share any information.
Right on. Thank you, Taylor. That's,
Cool.
big virtual round of applause for Taylor there. That was awesome. Yeah, fantastic. Thank you, guys.
Thanks a lot, guys.
Really cool. We will move on to James next, who's going to kind of continue this theme of thinking about, okay, well, applications, when you don't just have a typical deployment of what we've been talking about, this kind of like 1 day time scale. How do we use, and with camera tags and all the works, how do we start to adapt some of the CATS tools for that? So with that, we'll turn it over to James, who's one of my good colleagues for a long time. With James and I have, this is a picture of James and I tagging whales together. So we've done a lot of work together over the years. James is a PhD student in the Goldbogen lab with Jeremy Goldbogen, and has worked with Cascadia Research, who will transition to John Kalmakidis next. So I'll let James take it away from there.
All right. You guys can hear me. I'm sure you can see my slides. Dave just mentioned that. Just wanted to thank Dave and Will for putting this all on, and Taylor, that was awesome. Tough act to follow. So I just wanted to start by saying that I actually came into this field doing what you see in this picture here. So from that tagging perspective. And it wasn't very long ago that MATLAB was completely mystifying to me. So just want to say, hang in there, it gets easier and perhaps even fun as you move along. So early on, I started working with John Kalmakidis at Cascadia Research and collaborators at Moss Landing Marine Labs to try to increase the duration of deployments on baleen whales. And so in a perfect world, a suction cup attached tag would be able to answer all of our questions. But as we've all learned over this past week by identifying tag slips, we know that's just not the case. So in this project, we tested different methods of deploying the tags, as well as different types of attachment to the animals. And we were able to follow up with many of the animals to determine the impact of these tags over time. And the result of that project was the development of what we call medium duration archival tags. These tags incorporate either Wildlife Computers TDR10s or an AccuSound acoustic tag, depending on the types of questions you're trying to answer. These tags are attached with short darts, and they allow us to obtain deployment durations ranging from several days to multiple weeks. And so they sample high resolution data multiple times a second, collecting information about the environment, such as temperature and light level, as well as the animal's depth and fine scale movement metrics like pitch, roll, and speed, like we've all been calculating over the past few days. And this allows us to determine individual foraging events, or lunges, using the process that Sherelle demonstrated yesterday. And so these tags bridge a critical data gap, taking us from the dive scale to the scale of daily movements. And when these fine scale data are coupled with GPS locations at nearly every surfacing, it allows us to observe what the animal is doing and where. For example, over 17 days, this tag recorded 3,600 GPS locations where the animal utilized an area of over 850 square kilometers. And in that time, it completed 2,700 dives and over 6,000 foraging lunges. So the single day plotted above is actually just the last day of this deployment. It's shown in the bottom right. And it's also shown by just the red locations in the map to the bottom left. So since 2014, we've deployed over 100 of these medium duration tags on several species, including 28 on blue whales along the California coast. This has resulted in over 260 days of fairly high resolution data. So I'd say mission accomplished on the tag attachment improvements. And this did, of course, present some challenges related to getting these tags back, as Taylor mentioned. And as you can see from the map, I've had more than my fair share of trips 150 miles offshore or bouncing along Itapanga off Baja. But all this data also created some pretty significant processing challenges. So in raw data alone, this 266 days amounts to over 1 terabyte of data. And the import script for the tag I showed you earlier created a MATLAB file that was over 20 gigabytes. So the first thing I did when I started in the lab was to get my hands on a computer with a ton of RAM. But even with all the personal computing capabilities currently available, performing some of the complex operations in the PRH process sometimes took over a day. And the final straw for me came in 2018 when we had a tag stay on for over a month, and it produced a 75 gigabyte raw data file that needed to be processed. So that deployment forced me to reconsider how I deal with large data. So I'm not going to belabor you with more code since we've definitely dug deep this week, and it is Friday after all. But I'll just tell you that my solution was to this problem was to break the data into smaller 1 to 3 day chunks, depending on the deployment length. And in this instance, it resulted in 11 different partial data files. And so each partial data file is processed into a PRH individually, and I keep track of the tag slips and animal orientation from one file to the next, and I apply the end values of a file to the beginning of the next. And it really makes it a whole lot more manageable, even though it feels like sometimes I am doing a whole lot more PRHing, which, yeah, just takes a whole lot of time. And so once I have all the partial PRH files created, I then combine them all into a single PRH for further analysis. But you can see that this still results in a pretty massive file. I'm not claiming this is a particularly innovative solution, as it's mostly just a divide and conquer tactic. But what I think it does illustrate is that these tools can be adapted for a variety of applications. Indeed, it's worth noting that neither of the tag types we're using for these medium duration deployments are even CATS tags. But of course, once you're able to import the data into the same format, you can use the tools all the same. And so we've been working hard to make it compatible for multiple tag types. And indeed, I've used these tools for this purpose. Also, while this may be a rather unique problem to have, having too much data, it's not hard to imagine other applications where this type of issue might crop up. So for example, those of you working with species where a more durable attachment is possible, like tailor or in the sharks, you might experience the same thing. Or it may come as sampling rates or battery capacities increase, or as suction cup technologies improve. And finally, you simply might not have access to a powerful computer and might want to limit your exposure to sort of bogging down. So with these things in mind, I just have a few other practical considerations to working with large amounts of data. As I mentioned earlier, we have about 1 terabyte of just raw data. So for me, having a folder structure in place that allows you to separate raw from processed data is really useful. And I know Will was sort of getting at that and reiterating that earlier this morning. In my case, on my computer's hard drive, I use the tag data raw folder as more of like a work queue of files that have yet to be processed. And once I create the PRH and then move I then move all those raw files to deep storage elsewhere. I think this is also pretty applicable to those of you working with CATS video tags. Once you've created the data integrated videos, you probably don't need the raw videos clogging up your hard drive. And sort of speaking of deep storage and the elsewhere, from a practical standpoint, I use an amazing syncing software called GoodSync to back things up to a hard drive that's back on my laptop, which I'm sure many of you probably do the same. But also all of that data is then synced to the cloud. And for that, I use Google File Stream, which looks just like a hard drive on the computer and works pretty seamlessly in the background. And I'd say that it's been pretty critical for sharing data with collaborators. While all this is pretty basic, having a structure in place before the data gets out of control is really important. So just wanted to reiterate some of those points that Dave and Will have brought up over this workshop, is just having that folder structure early and just maintaining it is really useful, both for how you access things and just so you always know where things are. So, okay, so a few more practical matters. Getting back to MATLAB, in the early days, I'd always have the activity monitor open on my computer and just watching the RAM creep up as I loaded or processed data from my crazy tags. So it got me very clued in on the practice of sort of only loading what I need for a given task. And so this most recent version of the CATS process takes advantage of that paradigm. You'll notice throughout the main workhorse script that we've been using over the past few days that each section loads only what it needs and then saves what it creates. And so this paradigm allows you to pick up where you left off, which is sort of where that cell number comes in. But it also allows you to clean house between sections, which when you're working with large data is an especially good practice to employ. And so this brings us to, in terms of cleaning house, there's also some consideration with which file type in MATLAB you choose when you save your outputs. So this cropped up for me when I started working with these larger files. There are two file type versions that you can choose from. This version 6, which is the default when you save a file in MATLAB, or version 7.3. And so for any file greater than 2 gigabytes, MATLAB will throw an error if you try to save a version 6 file. And so that's what we're seeing in the try section of this code above. And then if it does throw an error, it then saves it as a version 7.3. Why, you might ask, don't I just always save it in 7.3? Well, there's two reasons. The first reason is that I can open and use a version 6 file in different platforms like R. You can actually open there's libraries to be able to open a version 6 file in R, which is a feature I'm sure most of you or many of you would appreciate. But something in version 7.3 makes that format unusable in any platform outside of MATLAB. So that's a pretty big limitation. Yeah, exactly, Dave. I say. The second reason is kind of a big one. It's that when you save something that could fit into a version 6 file as a version 7.3, it ends up being ridiculously large. So sometimes a 1 gigabyte file becomes 2 or 3 gigabytes, and it's just something that's incredibly inefficient with that file type. And use with caution, I would say, but it's become a reality for me that I have to pretty much save all of my files as 7.3. So I've been trying to think about ways to sort of steer around that. So this week's been a deep dive into taking raw tag data and converting it into standardized data for analysis. That's sort of the big thing that we're trying to do here. There's a high likelihood that the person doing all this work to clean and process the data will be looking to analyze it. But if it's going to be passed along to collaborators or used in publication supplements, some care must be taken. And so in considering large file types, it's worth exploring alternatives to the proprietary MATLAB data format. And so one alternative that Stacy and Dave both mentioned yesterday is the NetCDF format. I think even Will mentioned it today. So this is one of those unique examples where the solution to one problem actually solves instead of creates another. So you may be surprised to learn that the 2 gigabyte NC file here contains exactly the same information as the nearly 24 gigabyte MATLAB file. In fact, it contains even more information, which we'll discuss shortly. So what is NetCDF? If any of you are working with remotely sensed data, you've likely come across the NetCDF format. I think the most critical things to remember about NetCDF are that the data is self-describing and machine independent. So each file comes equipped with all the metadata that will allow someone else to interpret it without looking anywhere else. There's also libraries to read these files in R, Python, and MATLAB, and it works on Windows, Mac, and Linux. So it's pretty much, it's a really great format for sharing and making sure that anybody can use and open these files. So I'm learning to become a proponent of these files, and we'll see how we sort of look at that. So while this format was initially developed for geospatial data, like rasters and things like that, it's really adaptable for sensor data. And so Stacy and the folks working on animaltags.org put together some excellent resources for promoting the use of this file type for tag data. And I really encourage you to check out their wiki at the animaltags.org website. So with the help from our former postdoc Danuta, we've implemented these standards for the CATS tag and incorporated into the PRH process. And I think Will was just mentioning that. That's sort of one of the last steps of this last steps of this process. It's, I think, line 701 or 702 of the script that we're running there. In our implementation, sorry, pardon me. Yeah, so this allows us to convert the newly created PRH file that we create in this process into a CDF file, net CDF file. And so in our implementation, this process extracts the metadata about the deployment from the tag guide and then creates a sensor structure for each of the main variables of the PRH, all of which contain metadata about that data stream, such as the axis conventions, the sampling rate, the units, and whether it represents tag frame or whale frame. So the really cool part about this is that these sensor structures are compatible with the tag tools, all of those tag tools that Stacy was showing us yesterday. And also my lab mate Max has also incorporated them into some of the tools and packages he's developed in R. So there's sort of this, it's like a burgeoning direction to go on multiple platforms to be able to use these net CDF files. And I think, you know, just based on the file size and the fact that it's very portable and cross-platform usage is possible, I think it's a really good direction to think about, especially when working with large data sets. So finally, I just wanted to thank Dave and Will for putting this on and say that these types of workshops are such a huge help in making this type of research and analysis accessible to a wider audience. So I've had great mentorship along the way from my lab mates like Dave and Danuta. And I had the great fortune of attending a tag tools workshop put on by Mark Johnson back in 2017, which really opened the door for me and gave me the confidence to jump into tag data. So with all the help I've received, I'm doing my best to try and pay it forward. So I implore you to reach out if you have any questions. And if you are having trouble, just remember there's a community of people here that I'm sure are willing to help. So with that, I think I am there.
It's a great picture, James. Is that taken by a blue whale?
It was, gosh, it's one of those, yeah, one of those things, huh?
Cool. That's super awesome. I would just highlight as we think about questions, if you guys have questions for James, that one of the things that James was highlighting there is just a really cool aspect of what we're working on here is that James and Danuta and James said, okay, you know, this process will be better if we add in this script that will make a net CDF file. And so James and Danuta worked and came up with this script that would just throw into the CATS PRH file and made the process better. And so if anyone, as you guys are working through these tools, as you gain comfort in MATLAB, or if you're a pro already, if you're like, okay, well, this process is like a little clunky, Dave, let's make this better. I would be loved more than anything to throw in a new aspect, a new tool into this workflow. So please feel free to kind of keep improving these and adding these on there. I'll just open it up for the floor to anyone who has questions for James. We can always talk to him later on as well.
I did end up sharing my screen here, and I'm just showing you the result of a net CDF file. The left side is the actual PRH file that we created in this example, and I've gone through and run the CATS-NC script to create this net CDF file, and you end up with pretty much all the same variables. And the main things to make note of are that it by extracting all the information from the tag guide that we have here, this is all of the metadata about that. We end up putting all that information into the info field of this net CDF file, and you end up with a whole lot of data about, you know, the metadata about the deployment itself, you know, and then within each of the different variables like AT, you end up getting things like the sampling rate, whether or not it's regularly or irregularly sampled, and then the units. So it's just a really cool thing that all of the different metadata about the tag deployment travel with the tag data, which, you know, down the line, you know, if you share this with somebody, they'll still be able to use it. So I just think it's really cool.
James, one question: can you repeat what was the actual value that you needed to do the switch, the size file that you needed to do the switch for the version 7.3? Was it 2 gigabytes?
2 gigabytes, yeah. And I don't know. I mean, you know, we were seeing the example we were working with was a very short deployment, but I don't know how often you arrive at version 7.3s in your PRHing process, Dave. But you know, I get it. Every single file I create is basically 2 gigabytes or greater. So.
There you go. Yeah, really, we have that, you can see that format implemented a number of times in the script. Specifically, a lot of times we run into it in the audio files. So you see that early on in the make movie time scripts and some of the large data file creation process.
Yeah, and it does, I don't know why, but it's a pretty inefficient file type and it seems to be pretty bloated, but you know, avoid it if you can, but there's reason to use the 7.3, or there's.
One more long duration question here, James, for you. What are your typical sampling rates for the accelerometer on these long duration tags?
So we have, the TDR10 has a maximum sampling rate of 32 Hertz. So we end up, you know, yeah, so we're sampling that's the highest rate of that one, but the AccuSounds we run between 100 and 400 Hertz. So those can get to be quite large, but they're also limited by how the storage capacity on them as well. So it all pretty much evens out to about this, you know, 10 to 20 gigabyte files. Yeah, and then yeah, one thing I wanted to make note of, David mentioned earlier that jiggle speed, maybe the lower limit, he had mentioned was around 50 Hertz, but we are actually able to use jiggle speed using these 32 Hertz tag. And actually a paper just came out with using this TDR10 to actually identify calls on a blue whale. So that came out in Current Biology recently. So it did show up in the speed sensor when we were first processing that tag. We were able to see the calls as little spikes in the speed. And that sort of clued us into the fact that we were picking up calls on the accelerometer, and that led to that paper.
Blue whales call really low frequency. One more question on NetCDF, and then we'll move on. NetCDF does indeed also work back in MATLAB, so you can use that as kind of a typical format if you wanted to do that. So NetCDF is used often in the animal tag tutorials to actually just have that structure in there. So there's a lot of tag tools kind of designed around using that NetCDF structure. So after you create your MATLAB PRH file, you can just make that NetCDF structure and then import that in other places.
And I think from Jessica was asking about, is there a standardized metadata format? And that's all, that's what we've been using. And that's all from that animaltags.org website, as Danuta mentioned. So I would really encourage you to go check that out. It just gives you a really good framework. It's also, if you really want, there are, in the workhorse functions, there's a folder called structures. And inside that has a folder called reference. And so that has some of the reference of like data formats and structures of the NetCDF. That's basically just a copy of the website. But it's really useful, and it's helpful to sort of see how we can all get on the same page moving forward with data and coming up with a set of standards, which I think is really useful.
Awesome. A huge thanks to James for sharing those skills. You know, James has been thinking about this stuff a lot, so it's a good resource in addition to Will and myself about talking about some of these CATS tags. So thank you guys so much for jumping on here, James. It's really been good. Round of applause for James. Yeah, thanks. All right, we'll move on to John though. Next, we're going to talk a little bit more about different attachments and some of John's history doing some biologging work with a variety of different tags in a variety of different ways, and thinking about some of how and when and why to make decisions about how to put these things on animals. So with that, I'll turn it over to John Kalmakidis, who is the director of Cascadia Research Collective in Olympia, Washington, and has let him speak for himself.
Right. Can you guys hear me okay?
Yes, we hear you just fine.
Okay, perfect. Thank you. Nice to talk to all of you, and I'll share my screen here if that sounds good. And the title of my talk, I was going to talk a little bit about just a couple of different angles related to ethical considerations in tagging and some of the work we've been doing. I've both been very fortunate to work with Dave and James and quite a number of people that are in this workshop on different tag deployments. And for me, my career began a lot earlier, before many of these technologies were available, and then have been working more recently, learning new things, exploring the use of some of these new, both the new technology, new ways to attach them and have them stay. But The other part has been trying to see how the sort of long-term data we gather could be useful in assessing some of the impacts of tagging. I kind of feel like there are two elements. So calling it ethical considerations sort of encompasses both the fact that these tags can be very useful for conservation and management, but we also have to balance that against the fact that there are certain impacts that tagging has. And so I'm going to try to touch both sides of this, maybe a little bit heavier on the side of related to some of the impacts we've documented, especially of some of the more implant and more invasive tags that have been widely used prior to some of the suction cup and archival tags. So, in that, first of all, and I suspect you've heard about a number of these, so I'm not going to go into any detail on these, but just pointing out there are numerous valuable ways that tag deployments can inform, conservation protection of especially endangered species. Some of the early satellite tags that got position data were extremely valuable at identifying, habitat use, critical habitat areas, migration patterns, of endangered populations. More recently, we've been focused on using these tags to look at things like ship strike risk, which includes, how do whales respond to close approaches of vessels that might result in a ship strike, identifying do they react and move, and what does that tell us about what sort of mitigation strategy might be effective with ship strikes, helping us to identify areas of use and overlap with shipping lanes. I think I'll have one slide later about looking at the differential vulnerability of some of the large whales at night versus day, which, the nighttime behavior is a piece that we, in the past, prior to use of tags, didn't have much of a way to get an insight on. And so much of data on whale distribution comes from daytime sightings, and tags finally give us an ability to look at nighttime behavior and movements. We've also extensively used these tags, and I think you've probably heard about this throughout in this workshop, in behavioral response studies, particularly to different sounds like MFA sonar or ship noise. We actually have two other proposals to look at two other sound types and looking at how whales respond to them. And then more recently, and again, another one of the workshop participants here, looking at how tag data might be useful to both look at movements of whales and their use of the water column related to entanglement risk. So I want to recognize there are many aspects of these tag data that can be useful, and most of my work is focused on how we use them in conservation. But I also want to focus on some of the impacts and ways in which we also, in some cases, need to evaluate the impact of our tagging activities. And the need part sort of gets at some of the protections and permits that exist. And for example, in the United States, to do any kind of tagging work, you need a scientific research permit. In our waters, that's controlled by National Marine Fisheries Service. Those permits require you to document and make requests by species, locations, number, and the type of takes that you're going to take. And when I say type of takes, that includes, the degree to which you're going to, the degree and number of animals you would disturb, alter their behavior in any way. And that has to be part of the permit process. And in that permit process, you have to identify measures you're going to take to reduce impact, demonstrate the work is bona fide research, it's really needed and not duplicative. There's also a separate process that you have to go through, and that's the IACUP, the Institutional Animal Care and Use Committee is what that stands for. And basically, that's part of the Animal Care Act that is generally set up through individual institutions. It's a committee that exists within the institution conducting the work, and it focuses on individual animal care and impact, and includes things like pain or discomfort and suffering. And rather than looking at impacts on the more population level, it's actually looking at it at the individual animal level. Cascadia being a small organization, it was a bit of a challenge, but many years ago we formed our own institutional animal care and use committee We were fortunate to have people like Stephen Raverty, one of the most experienced, veterinarians working with large whales, as well as Francis Gulland as a part of that, and Alex Zerbini as our kind of statistician. But it also needs to include people from outside, both our institution or collaborators, and even outside the world of science. We actually have a psychologist on board and someone that works with media relations to look at it from other perspectives. So just so you're aware, before you go into tagging, you need to be concerned with permits. You need to be concerned with IACUC processes. Now, I'm going to show you a few slides related to different types of impacts and how we've assessed it. And before I do that, I'm going to show you a couple of, and I'll just point out, these are not typical examples. We don't generally go out and put many tags on the same animal, but I've grabbed a couple of examples where that was the case, and you actually see in these two examples, many of the types of tags deployed. On the top example, it's two blue whales side by side, and between the two, they have 3 tags on them, 2 suction cup attached tags. And you'll see at the back right of the top animal, a largely implanted Oregon State University satellite tag with only the very tip of the tag and antenna sticking out. And right below that image on the right, you actually see an early generation Oregon State University external satellite tag, but that was attached with very deep anchors that actually anchor into the muscle layer. So these are either tags that implant or have darts that implant through the skin and blubber and down into the muscle layer. And partly that's because it was discovered that tags anchored only in the blubber tended to have much shorter durations. You needed to anchor into that muscle layer to get longer term duration. But that's raised, consider that kind of higher impact tagging, because by going through into that deeper layer, you're also raising issues of any shearing effects that occur between the blubber and muscle layer, something that was looked at in a paper by Michael Moore from examination of a whale that stranded and had some of these tags and some needles that had been used to inject antibiotics and an anesthetic into the whale, and they discovered these shearing effects between the blubber and muscle layer. The lower animal there shows you some, a D tag up front, a limpet satellite tag attached to the dorsal fin, which is something I'll talk about in a second, and again, a suction cup tag. So obviously the types of impacts and ethical considerations can vary widely depending on the type of tag that you're actually using. Now, through a couple of studies that we've looked at, we've tried to look at use our follow-up studies of these animals to really look at what happens to these animals after some of these tag types have been deployed. A particular dramatic case that kind of raised the awareness of this issue to a higher level occurred in 2016 when a southern resident killer whale, L95, part of LPOD, that had one of these limpet satellite tags attached to it, which you see kind of diagrammed there and you saw in an earlier photo. It was found dead about 5 weeks after the tag was deployed on it. And the tag was no longer present, but an examination of the animal, and this went through both a detailed necropsy, there was a veterinary panel that reviewed the results, and I've just very briefly summarized the conclusions of these different groups that looked at this in detail, that the animal had died of a fungal infection, and the tag site was the likely source of that fungal infection introducing it. And while the tag was gone, the wound was still present, and some of the titanium petals that were part of that dart attachment had broken off and were still embedded at the tag site and had potentially contributed to keeping that wound open and the spread of that fungal infection. The animal was also in poor nutritional condition, and that likely played a role in its susceptibility to the fungal infection, because this fungus wasn't a particularly rare or unusual fungus, and generally in cases like zoo settings, it's primarily related to animals that are in poor condition, that are vulnerable to this type of fungal infection. Coming out of that event were recommendations to more thoroughly sterilize the darts. The particular tag that was deployed had actually been fired and missed, so it had come in contact with the water and had only been sprayed with alcohol, not fully disinfected prior to being reapplied. So there was a lot of focus on that. I tended to feel like we were maybe missing the point there, because these tags fly through the air, they contact the external part of the animal, and then push things that are on the surface in, so you can completely sterilize the tag, but that's still not addressing the fact that you are, when it comes into contact with the whale and pushes into the whale, you are going to be exposing things to that wound. There have been recommendations, and we've adopted in our own IACUC to try to avoid tagging animals that are in poor health or condition. And there also was a series of workshops on the development of best practices for tagging guidelines that was partly motivated by this incidence. And that's something that's available and now published. Now, Dave, I have a series of slides on some of our follow-up studies of the OSU tagged animals, and I can go through them pretty quickly, but maybe you want to tell me my time guideline here before I jump into that, and I'll know how quickly to go through those.
John, we've got until about 11:45 for this plus questions. So if you want to take another 5 or 10 minutes and go through a few more slides, that could be really useful for folks.
Okay, so I'll do this pretty quickly. These came out of a couple of studies that we've done, again, employing our follow-up, long-term follow-up studies with humpback blue whales and gray whales that we've studied for 30 years or more and had long-term histories. And we focused, for example, in one of these studies on some of the Oregon State University implant tagged animals that have been tagged going back to the 1990s, and for which we could match up the animal that was tagged with our photo ID. And we focused this on blue whales and the Pacific Coast Feeding Group gray whales, a group of gray whales that returns annually to the Pacific Northwest, and we have very high resighting rates. And again, I'm going to go quickly through this, but it involved looking at all our follow-up encounters with those whales, both to test what was the tag site condition and was there any evidence that those animals suffered a higher mortality rate. For this, we actually went through and we wanted control animals that represented untagged animals. So we actually used the OSU data, and where they had tagged an animal in a particular location, we went through and identified all the individuals that had been seen in that same area within, I think, about a 2-week period of that tagging event. And we treated those animals as untagged control animals, so we could compare things like survival rate between them. We had at least one case, and now we actually have a second case where one of these tagged animals washed up dead, and there was some ability to be able to examine and look at the tag site. Unfortunately, in some cases, like this case from 2010, it wasn't even realized that the animal that had died had been one of the tagged animals till after it was dead and buried. But we were able to look at photographs of the tag site. In neither of the cases was the tag similar to L95 necessarily implicated in the cause of death of the particular whale. One of the clearest cut cases we published in 2015, which was a blue whale with a long sighting history in California and the Sea of Cortez, the Gulf of California, that we could track. The black circles indicate just a similar pattern or marking you can see between all of these animals, and the black arrow shows the location of the tagging. And this was an animal tagged with one of those external tags, but with those long prongs that embedded into the muscle layer. And it turned out, it looked like one of these prongs had actually broken off and stayed embedded. A much more dramatic case of a part of the tag being left behind in the animal than was the case with the limpet satellite tag. And on the right, you see that the sighting history of this animal showed that while it was having calves in early years, after it was tagged, and especially after the swelling was first documented, this massive swelling that you see that developed at the tag site once that prong became embedded there. Once that swelling was documented starting in 1999, the animal had no calves. And it was only after the swelling had subsided in the late 2000s did we see a return to reproduction and calf. And so this seemed at least somewhat suggestive that we had 3 calves born to this animal in the period it didn't have a swelling, but this larger period where it had the swelling, we had no documented calves. So again, a little bit anecdotal because it was one animal, but it was our best case, if you will. You know, we could look at this various ways. For example, with the Pacific Coast Feeding Group gray whales, we could track the resighting rate by year of animals, both before the tagging occurred in 2009, and this compares in red our control animals and in blue our tagged animals. And you'll see that our control animals very closely mimicked the encounters in the years prior to tagging. So I think we had a good group of control animals. And then you can see there was some evidence of deviation in resighting rates that occurred after the tag deployment, with the tagged animals being resighted less often than the tagged animals. And that was attributable to a few animals that seemed to disappear. Bear in mind, the vast majority of whales that die don't get documented. So we know probably on the order of 95% or more of mortality, you never get documented with a carcass. So that's why we're trying to infer mortality from some of these resighting rates. I'll kind of skip some of these and just say that when we looked at survival rate and calculated it, the best models of survival rate, some of them included just a sex effect. This is what the Pacific Coast Feeding Group gray whales, and some included a tag effect. But it was difficult to conclude tagging was significant. A quick figure just to show you that when you looked at the models, you know, this is a model of survival rate that didn't include tag effect, showed in this case a lower survival rate for the females than the males. But the models that included survival rate showed a lower survival rate in blue of tagged animals than the red control animals. And these were from 2 of those animals, and you can see that's repeated in those. But again, we couldn't say there was a significant effect of tagging from this. I'll skip over blue whales. It was harder to do with blue whales. And from this, we saw, you know, the basic conclusions of the mortality rate was that there was some weak evidence of a possible tagging effect, but it was pretty weak. We did get much more data on kind of follow-up of what these tag site wound healing looked like, which ranged from, you know, depressions to small swellings to these fairly large swellings, you know, that you see here in the bottom right photograph. You know, but we were able to get good documentation of how these wounds heal and what that looks like. And again, I'll kind of jump to some of the major conclusions from that. You know, swelling and depressions were pretty common. I'm going to go pretty, I'll jump to these, like for example, swelling in gray whales. And we have that now blue and red are different levels of swelling. You'll see 0 is at the time of tagging. And you can see that the swelling typically occurred in the kind of in the months following the tag deployment, fractions of years, you know, and then generally decreased in gray whales, you know, and was generally gone by 3 years. In blue whales, you saw a little bit of a different effect, and this was driven by, I think, at least 2 of these animals that had remnants of the tag stay in them. And you actually saw some of this evidence of swelling increase over time, not see that rapid drop off that we saw in gray whales. Depressions, again, they formed early on, and we sort of documented the timeline of this. And I'm going to skip over that. Okay, so wound healing conclusions were just that, you know, we saw these evidence of swellings and depressions. They were more frequently in gray whales than in blue whales, but the most serious ones were these ones in blue whales and seemed to result from part of the tag remaining embedded. I do want to point out most of you are dealing with suction cup tags, you know, obviously much less invasive and serious, but I don't want to dismiss those impacts entirely. And here is one example that used a much stronger suction, one of these Canadian Tire suction cups, that then we actually saw evidence of it breaking the skin. You know, this was a suction cup that was on for multiple days, and follow-up showed that we actually had a wound that broke the skin as a result of suction cup tags. We've been focused a little more on the dart attached tags that James talked about. We've used these for some of the ship strike and day night differences. And I think the main, and this is, I'm down to my last slide or two here, Dave. The main conclusions is we've worked hard based on the conclusions of that study, earlier study, to try to find a dart material that doesn't break off and leave anything back. So we started out in 2014 using the same titanium darts that were used in L95. And these are in gray. And the main point I want to make that we did see some dart breakage. Quality 4 means one of the darts was actually broken. And we saw a pretty high incidence of this petal. These are these little metal petals that you see coming off the back of the dart here, where we saw a pretty high breakage rate of those petals in the titanium darts. What's led us to start using stainless steel darts. We saw a drop off in darts being broken and petal breakage, but we still had dart breakage and we still had petal breakage. The petal breakage was at a much lower rate. And then we started experimenting with heat treatments of these darts, and a heat treatment that would maximize malleability of the material, make it more bendable, and less likely to break. And we started with that heat treatment in 2017. And you can see in the 4 years since, we've had 0 dart breakage and 0 petal breakage, even though you'll see from these numbers here, we had still quite a bit of petals that would get bent even beyond 90 degrees, but they wouldn't break. So we consider this a major improvement. There's some debate that occurred within that tagging best practices. Some people feeling like, well, it's still better to use titanium. That's a more inert material suitable for use in implants. We felt that from the evidence of how leaving material in the tag can keep that tag wound open and result in these swellings, that better was using a material that reduces breakage. And we think we've succeeded, but there's some debate. The stainless steel we're using is less suitable for long-term implant in, say, humans than the titanium is. So the overall conclusions is, tags have these valuable ways they're useful in addressing important conservation issues, but we need to balance it with the potential impact of these tag deployments, especially we're using more invasive tags. And that this is both the responsibility of those of us conducting the research, but it also needs to be addressed through these permit and IACUC practices. And there is this best practices document out there that has now been published. I worry that I'm not sure it was produced heavily by some of the people deploying tags. I'm not sure it represents particularly strict guidelines, but still it's something that you should be aware is out there. Okay.
Thanks.
That's the end of my presentation.
John, that's fantastic. That's really useful, really cool context, really awesome to put out there. I have a number of questions here that people have been asking me that I'd love to throw out to you, and we'll open it up to the floor as well for people. And I also have Ari Friedlander back here on the line, as well as James is still here on the line as well. So these are three colleagues here who are some of the most experienced people, at least working with cetacean tags, and some other animals as well. Ari's done a lot of work with elephant seals as well, can speak to that a little bit. So we'd like to open up to some questions about these kinds of attachment methods and stuff. And I think one of the first 2 questions I'll have just to kind of open up the floor here. John, it sounds like, well, first of all, give them context. When people say tagging, they obviously mean a lot of different things. That can mean a lot of different things to a lot of different people. And so, when you're talking about these things, to be careful of, be clear about what we're actually talking about and what we're talking about the effects of tagging. What are we talking about? Physical effects to the animal or behavioral effects, and what kinds of tags do you actually mean for that? And I think one of the things you brought up, John, is the reason you could present this great data for us is that you do a really good job, and Cascade does a really good job of documenting the effects of the tags that you put out there. So I think one of the takeaways that I got from this is to whenever you're putting out tags and animals, to be able to document as much as you can. And then the question is, what other recommendations do you have really for tagging cetaceans or animals in general, in terms of like thinking about what are your best practices? How do you make the decisions about whether it's okay to put in a deep implantable tag or whether to use a suction cup tag.
Yeah, no, and it's it's a debate that we all need to have, and I think we draw the line in different places. So one of the debates that came up in the best practices, for example, was, well, a highly endangered species. Well, that's a case where it ups both sides of that equation. Suddenly now the information is even more necessary for conservation, but any impact you're having on the population now becomes even bigger and more significant to it. So it's kind of funny, both sides of that equation can vary with both the degree that the species is endangered. So I don't have any answer to it, other than, if your answer is always tagging is warranted, then maybe you're balancing too much one side of that equation. If you find yourself saying, no, we're not going to use that type of tag in this situation, then you're probably actually finding that correct balance. I don't know that. There's some people I feel that I see say they make that balance, but I've never seen them decide not to tag. But anyway, that's a way to to maybe view that you're taking some critical evaluation of that.
And maybe you could highlight again, just for you, just in your specific example, you do a lot of whale work. You and James are out on the boat, and you guys have 5 different tag types at your disposal to put on at any given time. What kinds of criteria what kind of decisions do you make to help you make those decisions to say, like, yes, this is an appropriate animal or not?
Right. Well, I'll give an example where we decided not to tag in a broad sense. And Dave, you were involved with this. When we first started tagging the Sounders gray whales, and I don't know if any point you talked about, these are gray whales that feed on the bottom, and we really had a desire to look at their feeding dynamics. So we initially deployed suction cup attached CATS tag, and it yielded great and exciting results, but it had been very much my plan to then deploy medium duration, dart attached tags to get a longer term picture of those animals. But the video tags and the CATS tags quickly revealed how much body to body contact these whales were having. Some of our tags got damaged and broken. We also got some incredibly long deployments. I think a 67-hour deployment, because we even saw in the video, sometimes the animals were rubbing off the tags, but other times they were pressing them back on by the contact they would have. So the tags were getting reattached. But we quickly realized, with the level of contact those gray whales were having with each other, imagine what that would mean if it was a dart attached tag and the type of injury that could result in. So that was a case where we made the decision to change our plans. And as much as I would have loved the data from the dart attached tags, in that particular setting, it looked like there was just so much contact, we decided against it, and that the value wasn't warranted. I'm not saying I'd ever not, we would potentially attach it in other settings, but we would have that kind of higher standard or concern about, are we sure the value we're getting can't be gotten some other way that had a lower impact. Other decisions in the field that you mentioned, James and I do go out with a lot of tag types, and I think we're often, trying to balance, the number of animals available, their approachability, the priority of the research needed. I would say a lot of those decisions don't necessarily involve that impact question, but sometimes they do. For example, with fin whales, within our IACUC, we've decided that fin whales, which sometimes can have a thinner blubber layer, our goal with our medium duration tags is to never use darts longer than the minimum blubber thickness of that particular animal, because we're not wanting to go through that blubber muscle interface and anchor. I think that does elevate the level of impact of the tagging. So with all our deployments on fin whales, we use much shorter darts, and that means shorter durations. So that's when we have the suite of tags available to us, that's one of the things we consider. Oh, if it's a fin whale, we're only going to deploy with short darts or suction cup. We're not going to use long darts. So that's one other in the field decision we make. And maybe James or Ari can jump in with any other insight, but those are a few thoughts I have related to that.
I think one thing I would add, John, is that often you're looking at the body condition of the animal and looking for, are you seeing vertebra or the shoulders there, and just making sure that it's in good health, and also skin condition. It can be a really big tell. If it has a high parasite load, you might choose to maybe, I think you would choose to not work with that animal for that reason. But I'll let Ari speak. Sorry, Ari.
Yeah, I agree with everything John said, and looks like Sarah Fortune made sort of a really interesting comment there as well about how you potentially do bias your sampling and your information by only selecting certain individuals to tag. And I think to me that speaks to this bigger question that John kind of alluded to, is that I was very resistant to use implantable tags for a long time until I felt comfortable that I had a research question that required that kind of data. And so initially I was interested in a day in the life of a whale and its very fine scale things. But then as the research kind of matured and these questions about longer term behavior and movements were things that became prescient for what we were doing, we sort of had to tackle that. And one of the ways that I found to do that was to work and collaborate with the people in the field who had demonstrated sort of that the use of those tags in an appropriate way and in a way that I was comfortable doing it. And that's one of the reasons I work so much with John and with James too, is that this development of what they're talking about with this medium duration tag to me is the exact situation where we've now increased the capacity in the tags so that we can actually create a tag that gives us information over a longer term that asks and allows us to answer questions that are really relevant to us. And so that's, in my mind, how I kind of think about the process of going into which tag type do you use for which different kind of study.
That's a good point. And Dave alluded to this. I'm just going to emphasize it, that all of these follow-up studies, are reliant on getting good photo identifications of the animals tagged, and trying to partner, Ari mentioned partnering with different groups, often in areas that you might be working. There will be, groups that are doing long-term photo ID tracking of the individuals, and partnering with them and making sure you're getting the type of photographs of the animals that will allow them to be tracked in the future were key elements. One of my big frustrations was that it only ended up being for about a third of the blue whales that had been satellite tagged, especially in early decades. Was there a suitable image to photo identify that individual? That improved dramatically later as the value of that was realized, but there was some major opportunities lost. From not getting that early on. Sometimes that means getting it before you deploy the tag. Sometimes it's the videos and photographs you get as you deploy the tag, and potentially a skin sample at the same time. But it also means sometimes investing the time after you deploy the tag to re-encounter the animal and get those good images. And that really helps for fixing where exactly is the tag located. So in follow-up studies, you can say, oh, this animal has a wound. Well, is that really where the tag was, or is that unrelated to the tag? And so getting that follow-up that shows the exact position of the tag is also essential then for interpreting those later photos.
John, Mark has a question to ask you. You showed you said that there was a best practices for tagging document. A, it would be great to get a link for that. Yes. And B, what does that document, or is there another document that addresses the idea of during a tagging process, specifically in the wild, I think specifically for cetaceans in this situation, is there a document that kind of addresses best practices to avoid harassment of animals during the tagging process?
And I'm trying to remember to what degree in that best practices document we cover that. So I don't know how much that's in there. It's obviously an important consideration. And I think it's a great point Mark brought up, because certainly in our permits and in the IACUC process, we have to address that. And I'd be happy to share our IACUC documents, and they have lists of citations of demonstration of that. And and there are even people I see in your workshop, that have even done some of the studies of follow-up and response. People like Mason Weinrich, I think, did one of the papers we cite that relates to behavioral response of animals to approach and biopsy. for example. So I will provide a link. I was just looking at, I think it's likely on our website. Russ Andrews is the senior author on the best practices guide. And I believe, I think, yeah, there it is. It's 2019. It is available through our website. If you go to the Cascadia website, it's a 2019.
Yeah, I said that. I think James had just gave him the link in the chat. So the link is in the chat now.
Perfect. Excellent.
Yep.
One thing also to keep in mind with this best practices is that, and I think the paper that John's talking about does a good job of mentioning it. It's really specific to the species that you're working with, what those best practices are. And John mentioned like the work Mason did, and having that with humpback whales, a species that we work quite a bit with, was really good as a foundation. But so many of the animals that we work with, we don't have that backup or that background information on how they respond to these kinds of things. And so the anecdotes that John is giving on this follow-up is really kind of the foundation for where you kind of go back and use that information to come up with what the best practices are to minimize sort of impact to different species. But it's going to vary completely depending on the species, and maybe even by the geographic region that you're working in.
I think we're going to take just a couple more questions here. One of the questions coming in from Marina kind of fits into a summary question, and so we'll see what else is coming in. Is to what degree do you guys see the potential to integrate more kind of physiological sensors into these tags in general for all kinds of species? And then also, like, where do you kind of see tagging going? Like, where do you see best practices going from here? Like, sometimes people tag because that's what's available, and sometimes we can think about what's the best option. So where do we kind of see both for the physiological aspects and where could tagging go from here?
Yeah, and maybe I'll defer to some of the other people on that. I don't know how much you've covered, things like the heart rate and other tags, but obviously, there are some exciting developments that I think are trying to be explored, including by people in this workshop, that I think being able to reliably add heart rate would be an important element, not just for basic biology of animals, but as another measure of being able to measure response of animals to different stressors in the environment. So, that's one key one that I see being worked on, and that we're still not there with. We joked about, I was excited to catch the presentation on sharks and ingested tags, and we thought about, it'd be really nice to have a tag inside the mouth of baleen whales, and get some measures like that. But anyway, not knowing what you've covered, I'll maybe defer to others to talk about some of the other directions that this might be going.
And we can also ask kind of the last question too, as you're thinking about that, any directional questions. Maybe this is good for Ari too, is Carrie's asking about like any considerations for trying to come up with a global framework for tagging procedures, and what kind of procedures happen for international, when you're thinking about using tags in different countries and in different countries' procedures.
Yeah, that's a big question. I mean, just in general, as a U.S. researcher, if you're working as, to the best of my knowledge, if you're working in another country's territorial waters, you have to follow those guidelines. If you're working on the high seas or in international waters, you're governed by the Marine Mammal Protection Act, and you have to sort of follow those guidelines and be permitted for that kind of activity. Honestly, I think a lot of that comes down to your personal ethics and what you're comfortable with, and sort of who you're comfortable working with. Like I mentioned, when I started satellite tagging, it was in the Antarctic, and we collaborated with a guy named Nick Gales, who's I think one of the best people who did sort of the due diligence on the impact of tagging, developing tags that had the most or the least amount of impact to the animals. And they have a governing structure similar to ours with IACUC that was quite restrictive initially. And so I personally made that choice to kind of go that route. And I think it's honestly, it's sort of up to individuals how you want to deal with that.
Awesome. Carrie, did you have a quick question as you're coming in here? I see you.
Oh, I was kind of going more towards the when you're applying for permits in those places. I've been learning recently when you're like, oh, cite Cascadia or cite NOAA, the other countries can get sort of offended of, well, the US does this, that's great. And, I would prefer to have the more rigorous best practices and use them on board myself. But integrating those best practices is more of a global collaboration where permitting agencies in those other countries don't get offended by what the US is trying to do as the standard. I think it's more of a sociological, question, which is, I was kind of curious of like how you've tried to push, or if anyone else has tried to push tighter regulations or restrictions on yourself in places that have lower regulations, because just, dealing with it now and probably will for a while.
The one point I'd make is in having done work in some other countries, I would say it's actually a kind of a mixed bag. I would say sometimes, other countries have stricter regulations than the US does, and you actually have to do more. I think, to me, I kind of view at least the US regulations represent a minimum I'm going to adhere to. If you're going to a place with more lax, I'd always be nervous if I'm working in that area because it has less permits. That would make me nervous, because I think it is a strength of the countries that have looked at this and adopted often protective regulations, and those are good things.
Awesome. I think we'll kind of wrap up at that. That's a good moment to wrap up on. A really huge thanks to John and to Ari and to James for jumping on here and having this conversation, and to everyone who's participating and giving good questions here. Again, you guys can contact these guys if you have more questions and comments, but huge thanks, John, for going through those slides, and for you guys for being involved here. We're going to take a quick break after this. And so if you guys want to keep coming up with questions, you probably can, but we'll take a quick break. We'll come back. It's 11:57 now. So we'll come back at 12:05 to kind of for the exciting conclusion for the workshop. We're going to come back and do a little bit of MATLAB and folder structuring. So I'm going to get your MATLAB open, get your files downloaded from so you can ready to go for this last step. And we'll do the kind of the exciting conclusion of the partial video making. So we'll get there, come back at 12:05. Thanks, guys. We'll talk to you very shortly. Larry, thanks so much for John and everyone else.
Bye, thanks.
Sure. Thank you, guys. Good reminder. I have to listen to these at night. It's not always great. Okay, so we have one of these GitHub websites is on here. I don't know why it still wants me to just click on that. Okay, and so there's a number of different. He's got a number of different tools on here. One of them is a visualizer. We've talked about a number of different ways to visualize tools, and so this is an R app. And if you like using R and you like visualizing these tools, once you have that NetCDF format, that portable format, you can use some of these tools to visualize data. So you have a nice three-dimensional trace here of your data from that way. So taking this depth profile and this kind of zoomed in depth profile and all these accelerometer signals to recreate that pseudo track in 3 dimensions in R. So that's one option that he has on there. Another cool option that he has on here is if you go back to, so you have your GitHub website, you can look through all these contributions and all these different pieces. You have all this repository links in a lot of people's websites. And so you have a lot of different kind of way. GitHub is a really great way to share some of your research and put it out there, both for like review purposes and also for like people to share. And so Max also has implemented a jiggle script. So to be able to take that accelerometer jiggle and talk about how to calculate speed from that using R. So if you get to that step and you're trying to, one of the things Max is actually working on is kind of a whole implementable tag processing, CATS tools processing script within R. And as Stacy talked about, you come into some problems with visualizing large data sets and stuff, but maybe we can do something that kind of goes back and forth between MATLAB and R a little bit more for those of you guys who have had those MATLAB frustrations throughout the time. But these kind of scripts are available for you to kind of have these intermediate steps of like processing tag jiggle through R. So that website is the Fluke and Feathers. Check that out on the GitHub. Okay, so back to MATLAB. One of the things that we have in your MATLAB tools, you've probably noticed, those of you guys who are constantly refreshing your GitHub, which is always fun. You have noticed a couple of changes that I made to the overall structure. One is I moved some of the Excel files just up into the Excel template and files, so you can find these templates and stuff in that Excel templates and files folder. And that way you just have these main scripts here, these main workhorse scripts in here. And we've talked about a lot of these. We did calibrate cats as the kind of the first step, import cats data when you first get your data. We've been working a lot in the main cats PRH tools. There's two more functions that we have that we're going to go over right now and how to use those. These are two variations. I will combine these at some point. They're basically at some point MATLAB did this awesome thing where they changed how they read the screen size of your screen. And so instead of taking the actual pixels of your screen, they assume a 100% width essentially. And that allows you to scale differently on different size monitors. But for our purposes, where we want an exact resolution of a figure to match the exact resolution of a video you had, that caused some problems. And so we have a couple of different versions. I think this 2019 one is going to work for basically everything. I think it goes back to about 2017 actually is when you have to start using this one, but it depends on your monitor and screen resolution. So you might have to try Vids Data Wireless 2019 and then go back to Vids Data Wireless if you have an older version of MATLAB. So 2020 works in this 2019 one as well. So this is kind of one final script. It's not well packaged as the other ones. It's a lot of naked code in here. But it's got two cells, just like we've been kind of working in. There's the cell 1 here, which is the setup, and then our cell 2 is kind of our workhorse, like make these partial files. And I want to, what we're trying to do here is basically make these final videos that we've been showing you. This is the last step. Once you have your nicely organized PRH file and your PRH file and your movie times file are the two critical ones you need for here. And you have all of your videos. This is the program that's going to sync them all together to actually make those nice overall videos like the Minky Whale I was just showing you guys, the data synced up on there. And so it has a couple of steps. There's a couple of things you have to kind of do preemptively. One of them is you do need to have a monitor, either a monitor with really big screen size or a dual monitor setup. And then we'll take it'll flash a little bit more if you have a dual monitor setup. And you have to kind of check that in your screen resolutions here. You can kind of go to you're using a Windows machine, you kind of have to check your display settings. And you can open up this section and change your display settings. So you're looking for this resolution here. And you want a resolution, for the 2K videos, our resolution of a 2K video is about 1920 pixels wide. And so because we're going a little bit wider than that with our little data box, you need about 2200, I think, pixel resolution. So 2500, as big as you can get, basically. If you're working on a laptop, there's some laptops with really fine resolution, and there's actually sometimes a button in here to change your zoom. So sometimes your laptops, if you have a newer laptop, it has a really, it might be like a really, like 4,000 pixels on your screen. And then you can change your zoom to be 100% and everything will get really small, but that will allow you to run this script. And this is the kind of script that's going to run overnight. So you may or may not be able to implement this tonight, and I can talk about some variations with you. There's some other kind of switches in here I'll talk about as well. But we can always work through this. If you get to the stop where you have a PRH and all you need to do is sync your data and you're stuck, well, let's chat and talk about it so you can get you to this next step, because there might be some switches you need to do. But one of the things, if you do have to change your resolution on your screen at all, or if you're plugging into an external monitor, you got to do those things before you open up MATLAB. So you have to change your monitor, change your screen size before you open up MATLAB, because MATLAB will kind of default to whatever. When it loads, it kind of figures out what your screen size is. So you need to plug into whatever your external monitor is, or your TV, or something that has a lot of pixel resolution, and then open up MATLAB, and then you can run this script. And it's independent. You don't have to have anything loaded in the workspace to run this. Now, there are a lot of assumptions that this makes, so sometimes this will break. Like if you truncated your data really right to the end of the file and you have movies on either side of it, that might cause some problems. So there's some things we might have to switch up for your data if you have some funky aspects to your data. But if you have that kind of standard format that we did, and including this example data, you should be able to run this script. And basically all you're doing here is going into opening up this Vids Data Wireless script. You're going to run section. It's going to ask you for where your files are. So we're going to navigate to our folder that has our data here. I'm just going to copy this line here. put it into my script here. So I'm in my, it's asking you for, if you have a Mac, you can't see this, I know, but it's asking you for the video files from one deployment. So where are my video files? So I've organized these, usually, you know, they're kind of all jumbled up. I just organized them by type, so they're all together. I'm going to select all of my movie files, video files 2 through 18. These are the files we've been watching and looking at for this Antarctic humpback whale. Select those files. And then it's going to find those. So everything's a little slow on the zoom aspect as well. And then it's going to ask you for your PRH file, which is outside of our raw folder here is our PRH file. So this is the file that has all of the data in there. And then if you have your movie times file right here, it'll find that automatically. If you don't have that there, it's going to ask you to find that movie times file. But mine's right here, so it's going to find that movie times file. And it's going to select that PRH file, which has all of our data in there, and it's going to load those in there. So it's going to take a second to load these. Oh, and then it's going to ask you, okay, the other trick is it's going to ask you for a directory where you want to put all your partial files in. Now, one of the things to think about is that this is going to take up a lot of data. So I actually have a whole hard drive, a 5 terabyte hard drive I use for this purpose, because each one of these deployments might take up about a terabyte of data just for this, just temporarily. This is a 2-part process here. We're first going to make 15-second clips of small data files, and then we're going to stitch them all together to get that kind of more normal size file. So, you know, in the end, our files for this deployment are going to be like a typical movie size file. So I'm going to scroll down. I'll show you the actual folder with our final videos in here. So this is our final videos. And so each one of these is, again, a normal size file. It's 2 gigabytes for a video file. This is 778 megabytes for video file. And, you know, this is what we're trying to create at the end here. This is the same files you've been looking at, but now stitched together. We'll see if that's going to load or not. But the intermediate files are very, very big. So here's our same video we've been looking at with the data stitched on there. The intermediate files are big because each one is basically raw data, and it's going to stay like that until we stitch them back together. Now, again, this is another thing where if you can come up with a workaround for this, awesome. But this is the best practices that we've come up with in order to make those nice videos. Okay, so any directory I want to put the partials in, it needs to be a folder that has a lot of space in it. So this is my kind of main workhorse Drobo here that you can use. You can also have a separate hard drive or something on your space, something that has about several hundred megabytes of space, several hundred gigabytes of space available. And, you know, you can run some of them and then sync them together and then run some more. So if you only have a limitation of that, we can, you know, work around that and only do a few at a time, but it's basically it's gonna cause all that. So I'm gonna select in my main folder here, I'm just gonna select one of these files here, and it's gonna create what's called a partials folder to start collecting all these files in there. So it's going to run that. It's going to create this partials folder in there. And basically the first thing it's going to do is it's importing the data and trying to just basically kind of make some plots of the data. So you can kind of, this is the plots, the main plots that are going to be stitched onto my video frame. So you'll see these pop up here in a second. There they are, come. And just we've been looking at some of these video files, but just so you can kind of see what's happening, this is the pitch and the roll. The roll is red and the heading is blue. This is going to be our whale here. That's going to pop up once the tag is on the whale here. This is going to be where our little kind of dolphin orientation is, and that's something I definitely stole from animaltags.org, a little Mark Johnson little script to help visualize the orientation of the animal. This is the depth profile, and what these numbers are on here is this is the video number. So this is just kind of giving you an overall quick look of where the videos overlap with the depth profile. So if you want to see like, okay, deep foraging action, you're going to come to video 9. If you want to see, you know, what's video 4 about? Oh, it's just a lot of surfacing behavior in here. And maybe that's where the bubble net feeding behavior is happening or something like that. So you can kind of use this as a guide to like, look, okay, where am I going to look within my videos to find that useful, interesting information? Yeah, and you can see like the gaps in here, right, are going to be because there's a gap in the video. This is going to be an audio file in here. So there's audio file 6 that goes between video file 5 and video file 7. So those are kind of all listed out here. This is where how long those durations of those videos files are. Okay, so those are those pieces here. Now, what's going to happen next? If you're sensitive to rapid motion, you might want to look away. But so now we're going to run the next cell. I just advance to the next cell. I didn't change any settings or anything like that, but it's basically it's going to basically load those files in here, and it's going to start reading those files one frame at a time. So it's reading in a video frame, and it's going to start flashing this, and it's going to flash this for like a day. It's going to run this file for like a day as it starts to like try to do this. So you want to be using a computer when you're not, you know, like you're going on vacation, or you have a separate computer that does this processing, or something like that. You can keep using your computer as you do this, but you have to be able to give out the noise. So it's actually going through, and you can see that little black and red lines in the bottom left there, kind of slowly moving through kind of one frame at a time, like where's the video now? Where's the video now? And every time it's flashing, it's basically taking a screenshot of that image and syncing that up with your video file and writing that video file right in there. So it's right in that file in there. So we'll let this kind of do one whole 15-second partial. I set the duration of this up at the beginning for about 15 seconds. You can, if you're having memory issues, you can shorten that duration again, just like we did for the audio files, and just like we did for when we originally read in the timestamps of the videos. You can shorten that duration to 5 seconds, or we used to have these three-dimensional videos, the VR videos, and those take just a huge amount of memory, and we had to shorten our duration read to 5 seconds. But for most of these 2Ks, and when you do if you do 4K video too, you probably will also have to do that as well, is shorten that to 5 seconds, so that so your computer can actually handle the length of these videos. And it's going to create this file. And so once it stops flashing, what it's doing now is it's writing that video file. So in my folder, it created this partial file. And in this partial file, it created it took your whale ID from that info file, from that info structure in your MATLAB PRH file. And it's basically creating, okay, here's my video 2 name. And it's creating a file here that is the first 15-second clip of this video. And I'm going to switch screens here and show you what the end result is. I'm going to stop this process here as soon as it pops up here. I'm going to switch my screen, stop my share here. I'll show you kind of what it looks like when it's done. Switch this other computer. This is the workshop evaluation we'll have you look at later. So this is the partial file now after it's been populated with all those data files. So this is a different deployment. This is a couple days later on a different deployment. But it makes a folder for every video. And within that video file are all of these different 15-second partial files. And these, you can see that they're raw AVI files. They're just raw, basically just raw data in there. There's no compression involved in that. And so they're huge. Each one of these files is 1 gigabyte big, so that they're just these big files. I'm gonna go over and stop. So each one of these files is big. So this whole that's why you need like a 4 terabyte drive or something to hold all these partial files in there. So then you have all these partial files. Well, that doesn't do me much good. It's only 15 seconds long, and it takes forever to load. It doesn't actually load very well. It's very slow to load. But this is my video. This is everything synced up together at that high resolution. And so what you do, there's one more step for this. Well, maybe 2 more steps, but one more step. Let me stop this one flashing. There's one more step for this, which is to then sync these all together. And so there's a number of software online that can use to stitch together video clips. Some are better than others. Some I used to use a free one that I can't even remember the name of, but because it was working for a while, and then all of a sudden it didn't give me the same resolution, and it would always compress things to a worse resolution. The whole point of doing this partly and maintaining this is to maintain the overall high resolution of the original video files, because we want to be able to do science on it. We want that original video resolution as much as we can, so that we can actually do things like see really precisely what the size of the krill is, or what the size of the blowhole is, or whatever. We don't want fuzziness or gaps in that data, or anything changing in our sample rate or anything like that. We want to be able to maintain that high quality video file that we had originally. And so I use Adobe Media Encoder, which usually lets you run for free as a standalone. So if you don't have an Adobe license, you should still be able to run Media Encoder specifically. Eventually, it might ask you for a license after 6 months or something. I'm not sure how exactly it works out. So if you don't have an Adobe license, you might have to find another online free software. But I use Adobe Media Encoder, and basically all you're doing is I just use it to add in these video files. So I'm just navigating to that folder, finding here's that first video there, selecting all of the files in that folder, stitching that clips together there. So I'm just going to stitch those together from that one folder. And before I do that, well, I can show you this. So this is going to load in here. It's going to load into this environment here. And I'll go back to this folder so you can see what's actually happening here. The other thing that this does is it kind of looks for places where there's very short videos and stitch them together, because sometimes you have very short videos and no one needs like 1,000 videos together all at once to have no one needs like, 50 videos. It's hard to look through all those. So what we try to do is stitch together videos that are very short. So even this one, this is basically videos 2 and 3 stitched together, because video 2 is very short. It's only a couple of minutes. And so I'd rather stitch together videos 2 and 3 together. And so when you see those ones come out, it'll basically show you, okay, this is videos 2 and 3 together. So here's video 2 and video 3, both of them stitched together into one video. So it's still like the overall length of the video is only going to be about 20 minutes still. It's not going to be, I try to limit the size of the videos to about half an hour. And you can change all those settings within the script if you want. Those are kind of our default settings that are in there. We just kind of are consistent about.
Question. What's the frame rate of the output video?
That's exactly what I was trying to say, is that the whole point of this is to try to match your frame rate, match everything from the original video files. So match the resolution, match the frame rate, etc. So the nominal frame rate, as we saw for most of these cats videos, is 30 frames per second with some variations. And we're trying to stitch that back together exactly the same way as the original video. But the final video will be easier to process and can be read on more portable and more useful. And does that answer that question well?
Yes.
Okay. And then with the final step here, basically all you're going to do is tell it where you want to put that final video. And once you start this running, you can just add in another video and kind of queue up all of the videos that you want to play. And once you're done with that, you're going to have this set of video files that I'm just putting them in. I'm taking this, when I plot it out here, I just kind of spit it out right here. And you have this series of now videos that all say stitched clips. This is all of those videos from 1570, all from all of these now stitched together into this one video clip. Now my last step here is going to be that nice folder structure that Will and James showed you guys. I have this folder here, this video. So in my tag data folder, in my tag data folder, in my cats files, I have this tag data structure. This is the folder I want to put all my files into. I put in all of my files in here. So I have all of my files in here except for these videos. Now all I have to do is just drag these video files into my deployment folder. And then there's only basically one more step, and that's all of these stay like stitched clip on them, and that's whatever. That's just a thing, and it has the video number on there, so you can find what you're looking for. But we have one more MATLAB script to run, which will then change what this says stitched clip there, or if it doesn't say anything at all, it'll change it to have a timestamp to the start of the video, because often you want to know, like, what was the whale doing at night, or what was it doing the next day, or whatever, and you want to help you navigate. A spot in your video, in a time of some event, and you want to look for that. So there's one final script you can run after this last step. You're not quite done. One final cosmetic script you can run. So here we are in our MATLAB file. I'll actually open up this one. Well, either way. So we'll open up, rename vids. And so basically this is one last script that allows you to just rename the scripts. It opens up your PRH file and allows you to put that information in there. So we'll go to here with this folder with our stitched clips. I want to know the files I want to rename. So all these files that I just put in there with the stitched clip there, it's going to grab those guys. And then it's going to load in the PRH file, because you noticed I had my PRH file right in this folder here. There's my PRH file right there. So it's in this folder. So it looks for that PRH file, loads it in there, and then says, oh, okay, video 2. That it's using that vidnum and that viddn file and saying, okay, that file starts on March 15th at 15:01:01. So that's now the timestamp of this video. So it's just basically renaming these videos. And sometimes it's a really quick process, and sometimes it takes forever as Windows tries to copy the files over themselves. But there you go. That's just going to basically run through there, finish up your files. Now you're done. You're done. You did it. You got everything on there. That's the whole bonus thing. So you can try this overnight tonight. It takes some time to run these partial files, but that's the procedure. And so the goal is to get people to be able to do this with your own data. And so, like I said, Will and I will keep working on the scripts to get us up to snuff so that there's going to be fewer errors. We'll set our January 1st kind of deadline. We're going to send you guys some evaluation forms in there, and one of them, it has an opportunity to add on to our email list if you'd like to be informed about future updates. You can also always just check the GitHub for more information about what we've changed and look for updates, and that's the benefit of the GitHub as well. So I'm going to stop that share. We're going to turn over to Will, who will talk a little bit more about some of the folder structure and some of the navigating the GitHub. While he's talking, we'll email you guys all the links to the evaluation forms. But then right when Will's done too, I'll just post the links in the chat for the evaluation forms.
Yeah, so there's one question, Dave, about videos. If you have segmented video clips, how to make them all synchronous, or would there be any problems with them being synchronous?
So I'm just going back into my share screen mode here, because that's exactly, that's what this person, that's why we kind of like read in all the videos. We have that viddn variable that keeps track of the timestamps of your videos. And at the start of my video here, that deployment video, these are, well, so this one is a bad example because they're all synchronous, but the other one I showed you there's gaps in them, or even there's a gap here, right? So they're throughout the day. And that's what this strip kind of keeps track of, and each video is independent and treats its video independently. So it uses that viddn variable to find the times, to find where that video actually starts and ends, and syncs those all up. So you don't have to then always be thinking about where those videos start and end. So that's why we go through those initial make movie time steps. So you have a timestamp at the start of each video, so you can kind of do that whole processing step. So that's exactly where that comes from. So yes, the whole thing will be synchronized and accounts for variability in your video time locations.
Awesome. So another thing quickly. So Dave just mentioned that there's going to be sort of like a post evaluation or a post assessment, or actually there's going to be both. We will give those out to you in the chat, but I also put the links to those on the homepage of the website. So if you guys just navigate there, it should be right near the top. So yeah, that would be really helpful for us to sort of, see what we did well and see what was helpful and what was maybe a little less helpful if we do this again in some form.
Yeah, I guess just to say on those evaluation notes, exactly, like this is, like I said, this is new to us, this idea of teaching workshops. So we'd love to get your feedback, and we'll take that into account for teaching future workshops. And it's helpful for all of us to kind of see new models and stuff. So really, thank you guys for all being in, all your patience and such for these things. So really, it's been really fun for us and really useful for us to get this feedback on these scripts and things. And we'll hopefully keep trying to make those better. We'll also ask you in one of those forums, just like one of the things we'd like to do is we're gonna try to write up a paper that kind of then puts all this out in like a publishable format, so you can use it and cite it and all that stuff. And we'd love to acknowledge all of you guys, your participation in this workshop. So just click yes if you're okay with that idea. So thanks. I'll turn it over to Will for some exciting conclusions here.
Right. Yeah, so I'm gonna just go through, I think, a little.
All right, sorry, Will, I'm gonna interrupt one more time because I see the forms starting to come in. I want to make sure everyone uses the right forms. So I'm gonna go ahead and put them into the chat right now.
Yeah, people can be working on them as well.
Just so Jesse just put in one, but actually just use these two. There's two forms. One is the workshop post-assessment, which mirrors the pre-assessment, just so we have a sense of like how much you learned on the course. And the other one is the course evaluation. So use those two forms, those two links if you can, and then you can let me know if you have any problems with that. But we'll have those there. And then we will email them out to everyone as well, just in case people are not here in the live version. Okay, thank you. Go ahead, Will. So I'll stop interrupting you. I'm gonna mute myself.
No worries. Yeah, so I'm just going to talk through a little bit here now and just give like a little wrap-up presentation, and also go through some of the ideas that I have for sort of adding things to the wiki to make it a little more helpful. This week has been really, really good for me to sort of see what's on there, what's useful, what people still need, in terms of like Dave is saying here, setting up the file structure. That's a piece on page 8, but thinking that should really be its own whole page, separated out and really described in a little more detail. So that's one of our plans moving forward. So what I'm going to do, I'll just sort of start off with this little presentation here. And as we move through, I might jump out for a little bit. Yeah, so moving forward, this is just kind of an overview of a lot of the things that Dave just sort of mentioned. And we've had some questions on, are these tools going to remain available? Are the Zoom sessions going to remain available? So the GitHub repository is going to be online. That's our goal is for that to be like a finalized tool that people can go to whenever they need. And I will be updating that as much as I can to reflect any changes in the code. So that's kind of where Dave was saying that January 1st sort of pseudo hard deadline that we're going to give ourselves to make sure that all the changes in the code are sort of in there, and then the wiki really reflects that. Because again, this week, we've sort of changed the code a little bit as we've gone to sort of fix some of the errors that have popped up, and the wiki hasn't necessarily been able to keep up with that in real time always. So yeah, thank you for all of your feedback on it. It's been a huge help to have a lot of people come in here and stress test this for us, really. So yeah, in the final manuscript, if you are willing, we would love to acknowledge that feedback. So the website might be transitioned a little bit. So currently it's sort of the website for the workshop itself, and that might not maintain into the future. So we might just change it to sort of be like the broad overview page for this toolkit. And then from there, you can still find all of the resources you would need. It'll just be in a slightly different format. And we've sort of been thinking a little bit this week and also previously trying to figure out the best way to make larger data sets available to people sort of in perpetuity. Google Drive is a little bit unwieldy, and it's a little bit dependent on file sizes and what people's download capabilities are and things like that. So what we are planning to do is put the example deployment and all of the data that goes along with that, all the videos, all the CSVs, everything there. And we're planning to put that onto Data Dryad. And so that when this short manuscript comes out, that data will be available to you through a specific DOI number. But for right now, it will remain up on that Google Drive. Same thing with the Zoom sessions, the actual recordings of these sessions. We will maintain them on Google Drive as long as we can. So they will be up there, but at some point in maybe the far future, they might not be able to be maintained up there, or we can see how useful those are. If people really are using them a lot, then we can find a way to maintain their availability into the future. Yeah, and so like I mentioned here, and like Dave mentioned, goal is to have a short manuscript at the end, which would probably be in a methods journal that sort of details the process and gives sort of a broad overview, and then directs you to all the necessary links and all the necessary tools that you would need to actually run this process yourself or work with it on your own. And so just to lay it out a little bit, what's kind of on my mind, what's on the wiki right now. So I have a few little introductions. So one is sort of like how to download the data from GitHub, either how to do it directly or how to do it through GitHub Desktop. And it's just like very, very brief overview there. And then also we have a bit of an introduction on sort of how to set up the cats tag. I mean, everything from as simple as how do you turn on the cats tag, because it's not always the most intuitive, And again, this is sort of based around the CATS tags that we've been using. So if you have another tag type, that might not all be useful. But we have some notes in there too about the UI, working with the UI, so the different pieces of the UI and what those might mean. And then up on the wiki now, there's detail on the more intense processes. So how to do the calibration. I have some ideas on how to update that a little bit. And then really how to perform the entire PRH creation process from the data importation all the way through to that final PRH product and the videos hopefully into the future. Yeah, so again, still to come, general updates for everything that's up there right now. I still have to finish up the pages for the video processing that we talked about today and sort of that splicing. Going to put more information on folder structures, file types, sort of just like meta information so that you know how to work the process, not just what the pieces are and where they are, but like, you know, if you need to name this file in a particular way, I can talk a little bit about why you might need to, you know, put a dot something after this or before, or a hyphen or something like that. And where that might be reading it into the code. And then finally, I really want to have a full list of the inputs and outputs from each step in the PRH process, because I think that's where a lot of questions were coming from. Of sort of like, okay, I'm running step 4. What am I supposed to have before I run this? And then what am I supposed to get out of it? And so I think having a little bit more of an idea of that, and not just what is supposed to come out, but where is that going to be placed? So like when the info file gets made, where does that get made? What folder structure is that going to be put into? The movie times file, same thing there. So understanding what's coming in and out of this script at each stage, I think would hopefully be really helpful to people. Yeah, so, and then I'm just gonna actually move over a little bit here for a moment to the website. So I put in a little, very simple forum on the website. And this is one reason why I would like to keep this up into the future. Having a small discussion forum here. So sort of just general discussion about anything related to CATS tagging or tagging in general, potentially. And then I've also just set up a little topic here on just, sort of if you're working through the toolkit, are there any errors that you're popping up with that you've encountered? And the idea there being that, Dave and I will be available if you email us and we'll do the best we can to sort of get back to you and help you fix the problems. But it might be something where if we fix the problem for one person, they might be able to go to another person who's having the same issue and help them fix it. So we want this to be a little more of a sort of a community resource that people can talk with each other and not just sort of rely on me and Dave to answer it, because maybe it'll take us a little while to get back to you or something, and we want you guys to be able to work through your data. That's really the most important thing for us. So that is there right now. You just need to go to the website and go to the discussion forum. It looks like there's some chat coming in. Are there any questions, Dave? Is it all good? Okay.
Yeah, you're all good. Just we're just talking about posting videos and stuff.
Oh, awesome. Okay, good. Okay, so all right. So moving on a little bit, I just wanted to sort of get into some of the sort of wrap up material here. I just want to thank some people again for helping us actually run this and make it work. So Mark Lammers and the Hawaiian Islands Humpback Whale National Marine Sanctuary Foundation. Mark was really instrumental in sort of coming to us and saying, hey, we're really interested in learning how to work with these CATS tags. Would you be willing to, run a little workshop for us? And that has ballooned a little bit into a larger workshop, but he was really instrumental in starting us on this. So big thanks to him for planting this idea. Big thanks to Jeremy and Ari for not only giving talks, but also giving me and Dave the freedom to take some time out and work on this. And then all the other members of the labs, both of the labs who have, many of whom have come and you've heard from this week, others who have just sort of been here helping out in the breakout rooms and things like that. And then all of our other guest speakers, everybody who's come and really shared their expertise on how they've been working with this data or pieces of the data or analyzing what comes out from these outputs. I think it's really cool to get that context from a bunch of other voices. And then thankfully for all of you guys for, Dave's really, I think said at the beginning of the day today, just your energy and coming in here and sticking with us and, really helping to make this fun for us and hopefully for you and really make it work. So that's been great, really. And then for any of our financial supporters for the Salinas High School internship program that we have at Hopkins, our GoFundMe is currently up to about $1,400, which I think I set our goal at 1,000 just because I wasn't sure. So the fact that we've surpassed that is really fun. It's really cool. And then we've had some donations from other avenues, including a very generous donation from Mark Lammers and the Hawaiian Islands Humpback Whale Foundation. So they've been a huge help on this. And yeah, so that's pretty much enough to fund a 1.5 plus of the Salinas High School program. So our goal is probably about $5,000 for funding 10 interns for a year. So this is huge. This is way more than I think we were expecting. So thank you so much.
Oh, it really is amazing, and it's a really cool program to do that. I just want to jump in on Will there, just because it's really special for us. I know we even had one of our former students was going to come actually give a guest lecture with us, but lives in a community where COVID is rampant and has COVID themselves. And so we did not want, so just a reminder that we're all pretty lucky to be able to have these workshops, these virtual workshops and stuff. And so this funding will help support people who are trying to learn these things without all the advantages. So really thank you guys for donating and for being part of this workshop to help do that. So, and if you do have funding for like trainings from your organization that could go to support this work, support this training, that would be really helpful for that. And so thank you so much for those of you guys who have already donated. That is really, like Will said, like a huge help for that. So really thanks for being there for that.
Very much. All right. With that, I think that might be the end of our structured material.
Thanks, guys. Yeah, it was really awesome. I see a lot of the great thank yous coming in. Thank you guys so much for being here. My partner's a teacher, and so she always loves that end of the Zoom workshop as well. I'm going to post, because everything's happening, I'll post the workshop links, the evaluations one more time, and we'll email them out as well, because we would love to give your feedback on all these aspects. So thank you guys so much for everything. Really great. We'll be around. Will, are you going to be there for the afternoon session today for any more last questions?
And
okay.
we actually had a question come in if we were willing and able to post the Zoom from those afternoon sessions, even though there's a lot of downtime. There is a lot of error checking that goes on in there that could be useful for people if they need it. So I think, yeah, we have those recordings, right, Dave?
Yeah. I said we'll post those.
Oh, okay. Sorry, I missed the response. Awesome.
Yep, all good.
Yeah, I'll get those links posted up later today.
Right on.
Awesome.
Thanks, guys.
Thank you all.
Have a great days. Hopefully we'll get to meet you guys all in person soon. So we'll keep it working. Let us know if you have questions as things pop up, and keep on logging, whatever it is. Right on.
Keep on catsing. Sounds weird. It doesn't fit.
Yeah. I always tell people to look up if they're trying to find the tags that we do. I just say like, oh, just Google cats videos. I'm sure you'll find it. You'll be right there. It'll be
I
really
must
easy.
admit,
First
I tried
hit.
that yesterday. Cats tag, nothing related.
Right on. All right. Closing the room. Get this recording posted. Thank you guys for all of being here. And if you have a chance to thank the guest speakers as well, if you know them or if you have their contact, please give them a thank you as well for those guys who were there today.
Very much. Yes.
Right on.